Hello and welcome back to PPMD's 27th Annual Conference. This is day 4. We made it to day 4. I'm Ryan Fisher, Chief Advocacy Officer at PPMD.
And I'm Eric Camino, PPMD's Vice President of Research and Clinical Innovation.
So we had another very full day yesterday.
Yeah,
very productive.
Very packed agenda. So yesterday we had some great talks on pulmonary management with both clinicians and patients. We heard from some companies that are developing products to manage inflammation, improve the mitochondria, as well as protect the heart. And that was for a wide range of Duchenne patients and Becker patients, non-ambulatory as well. We also had a fantastic presentation on gene editing, and we heard from FibroGen on their newly released data from their phase 2 trial. And you can find all of that on the conference hub. I'm going to hand it back over to Ryan.
Great. So we want to start by thanking our sponsors. Today's sponsor shout-outs are our elite sponsor, PTC, and then our platinum sponsors, FibroGen, NS Pharma, Pfizer, Sarepta, and Wayfair. Thank you so much for your support. Today's the final photo challenge. The category is anything goes. So show us people you love, your friends, pets, anything. Share your proudest moments, and we'll reveal the winner at the end of conference. And another reminder on the registry, if you register and complete all your surveys by 6:30, so the end of the month, you'll be put into a raffle for a free smartphone. So Eric?
Yeah, so coming up today, we are going to have some of our recently reported updates. We're going to hear from both Italfarmaco and ReveraGen. We're also going to have a look at some preclinical work being done in the area of gene therapy, as well as dystrophin restoration. And then finally, we're going to have some sessions on facilitating independence and quality of life. And with that, I'm going to give it back to Ryan to kick us off for today.
Great. So we have our first official talk. We are going to start today with recently reported updates from Italfarmaco on Givinostat trial for those with Becker. Joining us to share this new data is Dr. Paolo Batica. If you have questions, you can submit them in the box below your player. For now, I'm going to hand it over to Paolo to start the presentation.
So Ryan, thanks a lot for the invitation, and thanks to PPMD for giving us the opportunity to share this top-line result from our proof of concept study with Givinostat in Becker muscular dystrophy. If you can go to the next one, please. This is my disclaimer that you have seen already. I'm a full-time employee of Italfarmaco, which is the manufacturer of Givinostat. Givinostat is currently in development for the treatment of Duchenne and Becker. And as a result of that, it is not approved for sales in any country around the world, including the US. And this presentation is intended for dissemination and discussion of scientific information only. If you go to the next one, this is a summary of my presentation today. So I will start by reminding all of us the results we have already obtained in Duchenne with Givinostat. Then give you an overview of our phase 2 study in Becker muscular dystrophy, move on to the top-line results, and then conclude and tell you about the next steps. If you can go to the next one. Again, so I'll start with the summary of the results with Givinostat in Duchenne. Next, please. So Givinostat has been shown to counteract the muscle deterioration in Duchenne muscular dystrophy. We were actually able to demonstrate that at the histology level, so in muscle biopsies. This was true both in the MDX mouse, but also, and most importantly, in the Duchenne boys that actually took part in our phase 2 study. And after 1 year of treatment of Givinostat, on top of steroids, we were able to show that there was reduced fibrosis, reduced fat infiltration, increased muscle fibers diameter, and increased muscle regeneration. The kids then continued the study and are still on treatment now in their 8th year of treatment. And so we were able to confirm the result, observe the histology with MRI, and also to show that the long-term treatment with Givinostat is associated with a delay in loss of ambulation and a slower decline in the respiratory function. So given the similarities between Duchenne and Becker, and building on the experience that I just summarized, we decided to actually move on into a phase 2 proof of concept study to evaluate the effect of givinostat in ambulant Becker subjects aged 18 to 65, and who were able to walk between 200 and 450 meters at the 6-minute walk test at baseline. If you go to the next one. Again, next one again. This is a description of the study. The objective of the study was to demonstrate that givinostat can preserve the muscle and slow down the disease progression. And this was actually assessed by means of histology, muscle biopsies done at the beginning and at the end of the study, MRI, also this done at the beginning and at the end of the study, and then a number of functional tests that were done every 3 months, and you can see the list on the right-hand side. So after a period of screening, the patient that actually met the inclusion and exclusion criteria were randomized in a 2-to-1 ratio to givinostat or placebo. This was actually given as an oral suspension, and then the follow-up lasted for 12 months. If you go to the next one. This is the disposition of the patient in the phase 2 study. We screened 70 patients, to then have 51 that were randomized to either placebo or givinostat. And as you can see in the slides, the 17 patients that actually were on placebo completed the study. And in the givinostat arm, 30 patients completed the study. The reason for withdrawal are at the bottom of the slide. You can see 2 patients were actually withdrawn due to an adverse event. These were in one patient platelet decreased, and in the other one triglyceride increase, which is a signal that we have seen previously. And when the patient stopped the drug, the adverse event resolved completely. The 2 other patients actually had to withdraw from the study because they were, in essence, they had to travel from one country to another to complete the study. And obviously because of COVID, all the traveling were not possible. And so we had to withdraw the patient from the study. Next slide, please. This is a description of the baseline data of this patient. As you can see on the left-hand side, the age range between 19 and 61 years of age. On average, the patients on givinostat were younger than the one on placebo. If you go to the right-hand side, you see the baseline value in the different parameters that we measure. On average, we have approximately 25% of fibrosis in the muscle biopsy of the upper arm. The MRI fat fraction of the whole thigh, the fat fraction was close to 60% on average, so a highly infiltrated muscle. And then if you look at the function test, I guess the important information is to see that there was actually a high variability. So then if we look at the 2 groups and make some consideration about the baseline value, we actually had a significant increase between the groups, a significant imbalance between the groups in histology. So the randomization on histology did not work as well as we wanted. We looked at whether there were correlation between the different parameters at baseline, and the histology, MRI, or function did not correlate with age or time from diagnosis. This is something, by the way, that has already been shown in Becker. And then we also looked at whether histology and MRI were correlated with function and strength, and the answer is yes, they were highly correlated. And also this is replicating data that have been previously shown by others. If you go to the next one, then we move to the result. Next again. So this is the primary endpoint based on the experience with Duchenne muscular dystrophy, with givinostat in Duchenne muscular dystrophy. We selected total fibrosis as a primary endpoint. But as you can see in the graph, on the left-hand side, firstly, you can see that actually the fibrosis was actually larger in the placebo group compared to givinostat. So the two groups were in balance, as I said. But I believe even most importantly, there was actually no change in the 12 months. So this parameter did really not change throughout the 12 months. And as a result, also the two groups were not different. But if we go to the next slide, we can look at the MRI results that are actually telling a different story. So if we look at the fat fraction in either the whole thigh or the quadriceps, well, first of all, the parameters were actually, the fat fraction was balanced between the groups at baseline. And also, if you look at the right-hand side of each graph, you can see that there was a significant increase in fat fraction, so in fat infiltration in the placebo group. And this actually did not occur in givinostat. And as a result of that, the two groups were actually highly significantly different. If you go to the next one, we can see the results in the contractile cross-sectional area. So the fraction, the portion of the MRI image that is occupied by muscle. And also here for both the whole thigh and the quadriceps, the results were balanced at baseline, and there was actually a deterioration in the placebo group, which was not observed in the givinostat group. And this was highly significant in the whole thigh and was nearly significant for the quadriceps. So if you go to the next one, as I mentioned at the beginning, we did a number of tests, which are the standard tests being conducted in particular from the experience in Duchenne. Also here, we have the same situation as for histology. So there was actually no change over time in this test, the 6-minute walk distance, the Forster climb, the time 10-meter walk run, and the time to rise in either givinostat or the placebo group. This, by the way, was also true for the strength, where no change over time was observed. And also here, at the end, we did not have a difference between the two groups. If you go to the next one, the situation was actually different with the standing and transfer domain in the motor function measure, which is a 32-question questionnaire that is looking at different domains related to mobility. And as you can see here, in this case, we did see a decline throughout the time of this parameter. And if we look at the result at month 12, the difference between the placebo and the givinostat group, the decline in this group was actually slower than in the placebo group. If you go to the next one, we can then look at the safety. In the 51 patients treated in the study, we did not have any serious adverse event or death. The most frequent adverse events are the one that we actually I have shown other times in Duchenne. So diarrhea, platelet decreased, and increased triglyceride. We actually reduced the dose from approximately 70 milligram twice a daily, which was the original dose, to 50 milligram twice a daily to reduce the incidence of the adverse event. And we obviously measure a number of other safety parameters, including ECG, and we did not see any change in those parameters in the givinostat group. So if you go to the next one, again, please. So overall, we can conclude that the study clearly failed the primary endpoint, but we believe that actually this was due to the high variability of the histology parameter with a significant imbalance between the groups at baseline, and also by the fact that there was actually no change over time in this parameter. As I said, the picture is totally different with MRI. If we look at the MRI of the whole thigh and of the quadricep, we can confirm the ability of givinostat to counteract the muscle deterioration, also in Becker, by preventing fat infiltration and muscle atrophy. When we looked at the function and strength, we saw that actually there was no impairment or a very slow impairment, if any. And so this made it very difficult, if not impossible, to demonstrate a difference between the two groups. The picture was different for the standing and transfer domain in the motor function measure, where the results are actually suggestive of a potential benefit of givinostat. The potential benefit is also suggested by the fact that the MRI is actually highly correlated with function and strength at baseline. So we believe that the results that we obtain in MRI will eventually translate also into a functional benefit. And as I said at the beginning, the givinostat treatment also in Becker individuals continue to show a good safety profile. So if you go to the next one, these are really the next steps and also the relevance, obviously, to the ongoing evaluation in Duchenne muscular dystrophy. With regards to Becker, Italfarmaco will, on one hand, continue to evaluate the data. We have many more analyses to conduct, and then also to discuss with the European and US regulatory authorities to define the best path forward for givinostat in Becker. And obviously, with regards to the Duchenne, this was clearly a standalone study, but I believe that the results are actually confirming the beneficial effect of givinostat, and this, if you want, replicated what we had observed already in Duchenne boys. And with that, let me, if you go to the next one, let me thank, obviously, all the patients that are enrolled and have conducted our studies. Their participation has been, obviously, key to the development of this drug. Clinical trials sites have done an excellent job, in particular, if we consider the pandemic situation. And obviously, a big thank to all the patient organizations that have actually supported us. Let me also remind you that this study was funded together by Italfarmaco and by the Regione Lombardia with a grant coming from the European region. And at the bottom, you can see the place where you can find more answer if you want to know more about the studies or any question you might have. And with that, I'm happy to take any question. Thank you.
Thank you, Paolo. This is certainly a challenging population to work with where we don't have robust natural history data. And as we saw in your presentation, looking at something like total fibrosis over 12 months, there's not much change, but we did see some of that with fat fraction. And so kind of two questions for you. One is, as you think about moving forward, is something like fat fraction a preferred outcome measure to try and use? I mean, obviously, we do have to still work on it being qualified. And the other question is, we saw, again, 12 months, hard to see change. Is this a population where we have to think about longer trials, which can be challenging for families and for those patients? But would love to get your thoughts on that.
Eric, you have touched all the different questions which are running in my brain since I see the results. So I think you got them perfect. So let me start from MRI. I think MRI, these results are confirming the value of quantitative MRI. I think they really are a very important tool in our muscular dystrophies to look at the evolution of the disease in the muscle, which is a key tissue that is affected by these diseases. And I'm actually a strong supporter of MRI as a potential endpoint also for the future. And I think this applies to Duchenne, but also applies to Becker. With regards to the evolution, I think that the point, you said it quite clearly and quite rightly. We have limited history, natural history in Becker. And what we saw in these patients, so these patients were selected as patients that were, in essence, doing still fairly well on a 6-minute walk test, because they were walking between 200 and 450, which is not a bad value. But for example, if you look at the MRI fat fraction, I mean, they had 60% of fat infiltration. So there is probably these patients, their muscle are severely affected, but possibly they have better compensatory mechanism. And so we will have to think about how to best assess the effect of a drug in this population. And this is what we will have to do in the future and also discuss with the regulatory authorities.
Great, thank you. I think we'd also just like to hear your experiences. We're seeing some more companies as well look at Becker populations. In terms of recruitment, was that a challenge with this population, or were you was it something that was easy to fill in your enrollment?
Well, I think I have to answer to the question. The first one is that the number of patients that are willing to take part in the study is large, because as you just said, there is actually very limited clinical studies ongoing in Becker. Obviously, the issue comes when you start selecting what you believe is a more responsive population. As you have seen in our trial, we have, in essence, screened 70 patients to select 51. And by the way, there was probably an even larger group that was pre-screened. But I mean, given what we just saw and this very slow progression, I think we will have to identify which is the best way to recruit patients that actually can show some effect in a decent time. I mean, I don't think that anybody of us wants to run a 3 or 4 years placebo-controlled study. I mean, that would not be possible.
Yeah, no, I agree. Paolo, have those patients stayed on drug and open label? Any of them drop out?
This is actually not, we did not start an open label because, as I said, this was limited to the funding received from the Regione Lombardia. But obviously, this is something that we need to reason about for the future. Yes.
Sure. And there's just a follow-up question. I don't think that many U.S. patients are familiar with the motor function measure, or MFM, that's been used here. I was wondering about taking this forward with regulators. Have you had talks with regulators about that measure, and any comments in regards?
So this is actually a scale that is used in Europe. I believe the originator are from France. Mm-hmm. And actually, the EMA was keen for us to have it also in Duchenne. Mm-hmm. You are right, it's not that familiar in the US, and this will be a discussion for the future with the FDA. But I guess to me, the important point is that it looks like another type of questionnaire, which, let's say, looks more in a holistic way at the changes in the disease, might be better suited in Becker. I think it still needs to be confirmed, but these original results might suggest that.
Great. Well, we are grateful that you're studying this population, and we are here to support however we can. So thank you so much.
Don't worry, I'll come and knock at your door.
Yes, and thank you for helping us with the guidance. We know you're going to be involved in that process along with many other companies, so thank you.
Thank you.
Okay.
Thanks. Thank you. Okay, thank you.
Great. Great. So we're going to have our first Pat chat now. We're turning to Pat, who's joined by Annemiek Artemis for us to discuss gene therapy. Pat, I am going to hand it over to you in a minute, but I just want to say that Annemiek's a personal friend and somebody I admire deeply, and we're grateful for all she does. So, Pat, handing it to you.
Thank you, Ryan. And hello, Annemiek. How are you?
I'm sad that we have to have these virtual meetings still, but it's nice to be here virtually.
Yes, it is nice to be here, but we can't wait to see you in person very soon. So hopefully your world will open as well and we can join together. So Annemiek, we've had lots of discussions on gene therapy, lots of excitement and enthusiasm around gene therapy and the hope of CRISPR. I think one of the most confusing things for parents is when they hear scientific information on what appears to be several mouse models, dog models, rat models, and rabbit. So could you tell us a little bit about since you're a mouse doctor, could you tell us a little bit about what mouse, for what reason, and what to conclude from that?
So I think there is a lot of different mouse models for Duchenne. However, there's one that's mostly used, and that's called the MDX mouse. It's in a specific so it's a black mouse. It's in a specific background, and that's the strain that's been used by most people, and also the strain that's been used for over 25 years. What's interesting is that this mouse has a spontaneous mutation, so we didn't genetically engineer it. It's spontaneous. It has a spontaneous mutation of the mouse dystrophin gene, and what's good about this mouse is it has a mutation of the mouse dystrophin gene. Where this mouse differs from humans is that it regenerates really, really well. So it has muscle damage, like Duchenne patients, but the mouse compensates. So it regenerates. So for all the damaged muscle, it keeps regenerating. In the wild, that would not work because it would have to eat a lot. It runs less quick than the other mice. But in a laboratory, it works well because they get their food, there's no predators. So that mouse is useful. However, the muscle quality of that mouse is better than the muscle quality of patients. And therefore, whatever therapy you test in that mouse, you will always overestimate the effect because whatever therapy you use, gene therapy or other therapies, it always depends on the amount of muscle that you have. And this mouse has a lot of muscle. Now, recently, there's a new mouse model that's called the D2 MDX. And basically, it's the MDX, but now it has a beige, goldish, grayish color fur. It has a different background, so it just means it's a different strain, a different race of mouse. And that mouse has more fibrosis and less regeneration. So that's more similar to humans. However, there's also some issues with that mouse. And the first one is that we don't know a lot about this mouse because it's new. So people are now doing a lot of work because if you want to test gene therapy or whatever therapy in a new mouse, you first need to know, well, how does this mouse behave normally? You probably heard about natural history studies for patients. We need to do the same for mouse. For mice, we need to know when is the disease escalating, when should we start treating, when should we evaluate treatment effect. So for that, for this new model, we know a little bit, but not yet enough. And then finally, a mouse model that's very frequently used is the MDX utrophin double knockout mouse. So that mouse has no dystrophin and also no utrophin, and utrophin partially compensates for dystrophin. So this mouse is more severely affected. However, patients do have utrophin. So this means that whatever you're testing in this mouse, well, if you're restoring dystrophin, you know that the effect that you will look at will be due to dystrophin restoration. But if you look at downstream effects, you're never sure, is this targeting something that's going wrong because there's no dystrophin, because there's no utrophin, or because both are missing. So that means that it's really difficult to translate that to patients.
Wow. So, many studies are done in the MDX, and now we're seeing those models come forward. So, is there one of those models that better replicates the cardiac issues? We've all struggled with the question about the heart and its progression and what we can learn about it and know about it and certainly prevent it.
So, the D2 MDX mouse has a more severe cardiac disease than the regular MDX. However, it also has calcifications in the heart. And we see calcifications in Duchenne patients at a low level, but it's much more prominent in this mouse. So that's the challenge. So there is more cardiac problems. So with that regard, it's a better model. But the challenge is that the pathology is different in this mouse than it is in humans. So the risk there is, it may be possible that you treat something and that it doesn't matter because you treat the cause of the disease, and either it shows in calcification or it shows in fibrosis, and it will reduce both. So then you can translate from mouse to human. But it's also possible that you have something that only works on calcification. So then you're developing a drug for the mouse rather than for the human. So that's, again, we need to find out more. But it's really important. I think you need to ask the right question to the mouse. And so what is your question? And then you need to look which mouse model is best for this. And it's possible that sometimes this will be the D2 MDX mouse, and sometimes it will be older regular MDX mice, because they also develop a cardiomyopathy. So it depends on the question that you ask. There's of course also now a rat model, there's pig models, there's dog models, and sometimes another model may be a better model for your question. So of course the mice are easy because they're small and relatively cheap, but for some questions you may just need to go into, for instance, the rat model.
And why would you do a rat or a pig? Are there certain questions that you ask of rats and pigs and in rabbits?
So again, that depends on what pathology is recapitulated in these models. So for instance, the rat model has a more severe muscle phenotype to begin with. The dog has also a more severe phenotype, but between different dogs, there is a lot of variation. So that makes it really challenging. The pig has an extremely severe phenotype. So most pigs don't live beyond the first few weeks of life. So they die really rapidly, and a few will survive longer. So for some questions, if you look at body-wide delivery, that's much easier in a 20-gram mouse than in a 20-kilogram boy. So then if you use the rats, and they used to say that rats are about 200 grams, but I know that some of the Duchenne rat models, they can grow up to be 1 kilo. Well, that's closer to the 20-kilo boy than the 20-gram mouse. So I think it depends on the question. If your question is, does this work at all, then often the mouse is enough. But if your question is, well, I now want to see whether I can deliver this to all the muscles in the body, and you've shown in the mouse that you can do it, then the step to the humans is very big, because you go from 20 grams to 20 kilograms. So that's 1,000-fold difference. If you have the rat in between, and then you can also, you know better, like, okay, so if the scaling is different, if the volume of the muscle is different, how does that differ from mouse to rats? And then you will know, so likely if we go into humans, this is what we can expect.
This is so hard, right? This is just
it is, yeah.
So
and
hard.
I think the challenge is also that for each question, you have to find the right model. You will hear in my answer, there is not one answer. It's not like you always have to use this or you always have to use that. It really depends on the question that you ask. And there likely for each model there will not be, or for each question there will not be a perfect model. So you will have to find the best model. But some models really won't work for your question. So that's really, you have to be careful there. It's not a one size fits all. And I think also that's where some of these studies go wrong, where you have these really hyped studies that say, well, and this works in mice, and now all patients should be treating this, or we should start a clinical trial for patients. Well, if your model is not optimal, if your drug fully relies on having really good muscle quality, yes, it will work in mouse, but will it also work in human? You have to wait and see. And I think that makes it more difficult. And it is difficult. It's difficult enough in patients, but also in the preclinical space, it's difficult. But there's also now a lot of effort to try and standardize things. So to collaborate with each other, to provide guidance, like for which question do you need which model? And also if you use a model, don't only look at one muscle and see whether there's less fibrosis. This is another thing that worries me. A lot of people will look at the tibialis anterior, the TA muscle. Well, in mouse, that is one of the least affected muscle. And then you will hear people say, we see a 400% reduction in fibrosis. Well, that means that the fibrosis goes from, say, 2% to 0.5%. That is not that impressive. If they would have done it in the diaphragm, where it goes from 30% to, say, 7-point-something%, that would have impressed me a lot more. So you really have to also be careful. If you use the right model, which muscle do you analyze? Which do you only look at muscle, or ideally do you also look at function, etc.?
Well, I think standardization is so important here, because for you all who are doing the work at the bench to really find the targets that are going to be potentially useful to slow or stop this disease course. But I think for us on the outside, and we read an article that says exactly what you say, 100% expression in the tibialis anterior, we've got magic here. Yeah. And then it turns out, first of all, to be years and years before it comes to the clinic, and then turns out to be maybe not as magical as we had hoped in the first place. Yeah. So, Annemiek, we've seen antisense. We have 4 approvals in the United States. There are antisense going on. So it looks like that the proof of concept is here in terms of antisense oligonucleotides. We see microdystrophins, and we always wonder about we were talking this morning about the microdystrophins, and does it matter what the microdystrophin is because of the carrying capacity of the serotype? But is it more about how much you get in there, how much you get into the muscle cells? So I'd love for you to comment on that.
Yeah. So I think there's 2 aspects. So for the exon skipping, what we know, you get larger dystrophins than the microdystrophin. I mean, they're probably about 2 times larger or even more. And for exon skipping, we know that there is very low levels of these larger dystrophins. There's definitely room for improvement. I think everyone, also the companies that develop these exon skipping drugs, they also will agree there's room for improvement. We can do a little bit. It needs to be better. Now, for the microdystrophin, we see that in the clinical trials, you really see significant levels. You see good amount. I mean, I'm really pleasantly surprised about the levels of microdystrophin that people see. Not just the amounts, but also the percentage of nuclei. So I think that is really promising. But what we don't know is how functional will that microdystrophin be. And well, it's not a microdystrophin. I think there's now 4, because in Europe there's also Genoton that started the trial. So there's now 4 different microdystrophins, probably more in preclinical development. And they are very similar, but they differ a little bit. So that will be question 1. Are they equally functional? But question 2 is, of course, also how functional are they? With exon skipping, we know that these dystrophins are functional because we see them in Becker. Becker is really the proof that these dystrophins are functional. The smallest Becker patient still is that dystrophin is still 40% larger than the microdystrophin. So we don't know how functional it will be. I think that is now one question that people really hope to see. And well, of course, we now have the placebo-controlled trial, so hopefully we will also see that these microdystrophins are functional in slowing down disease progression. But we don't know because there is no Becker patient with a microdystrophin. And I think one other challenge is that the microdystrophin, it's not fully functional. So we know with time it will go away. And the big question I think now is, will it be functional and how long will it stay there? And of course we hope decades. But if it would be years, then of course that would be a problem, because then you have to retreat, and well, that's of course also a problem.
Yes, well, we are very hopeful, very enthusiastic.
Yes.
So maybe one day we can layer a microdystrophin and then right after that an antisense with a bigger protein. Annemiek, we miss you terribly. We really miss you. I hope to see you soon. Thank you.
I hope to be there next year in person.
Yes.
With chocolates.
Yes, with chocolates. Absolutely. You're the one. Thank you so much for joining us.
Thank
we'll see you soon.
Take care.
All right. Bye.
Thank you, Pat, and thank you, Annemiek. I could listen to her talk all day,
Yeah,
honestly.
she's fantastic.
I think every family should listen to these presentations. It's really hard to interpret what's coming out when it comes to what's happening in a mouse and then how it actually converts to a human. I think this information is so critical. So we're now going to move on to our next in the pipeline session on preclinical and early stage gene therapy products. We have a great panel ahead of us, don't we, Eric?
We do. So we're going to be joined by Matthew Pletcher from Astellas Gene Therapy, Olivier Danos from Regenex Bio, and Kevin Flanagan from Nationwide Children's Hospital. So as we go through these presentations, at the end we are going to have an open Q&A. So again, if you have questions, please feel free to submit them to that comment box below the live stream. And so with that, I think we're going to start off with Astellas Gene Therapy. So Matthew, whenever you're ready, you can start for us. Oh, sorry, we have the context video from Mena first, so.
The root cause of Duchenne is a variant or change in the DMD gene that prevents the production of the dystrophin protein. The absence of this protein leads to a disrupted and weak membrane that surrounds muscle cells. One strategy to correct this lack of dystrophin is to deliver a replacement gene to the muscle cells that can function in place of the missing dystrophin. This is called gene therapy. The dystrophin gene is large. Current gene therapy clinical trials in Duchenne are exploring the use of microdystrophins. The microdystrophins are smaller genes than the full-size dystrophin gene. They are based on dystrophin changes seen in patients with milder cases. The hope is that these microdystrophins can act as a replacement for the missing full-size dystrophin and produce the protein that is missing. To understand gene therapy, we need to look at the three main components used to deliver the therapy. First, there is the transgene. This is the instruction for the body to produce a protein. In the case of current Duchenne gene therapy trials, different microdystrophin transgenes are being delivered. Second, there is a promoter sequence. This is a piece of DNA that tells the body where to turn on production of the transgene. The gene can get into many different tissues, not just the muscle, so we want to ensure the new protein is only produced in tissues where there would be a benefit, such as the skeletal or cardiac muscle. Third, there is the vector, which acts as the transporter for the transgene and the promoter to get into the target tissue. In current studies, a modified virus called adeno-associated virus, or AAV, is used. The AAV is a tiny virus with a limited storage capacity, which is why current studies use a microdystrophin transgene rather than full-length dystrophin instructions. There are different serotypes or versions of the virus, such as AAV8, AAV9, and AAVrh74, that are being used in trials. AAV does exist in nature, though it has not been found to be disease-causing in humans. However, because it does exist in nature, it is possible for an individual to have been exposed to AAV and for their immune system to produce antibodies to prevent infection. If an individual has been previously exposed to AAV, they may make antibodies that would prevent the gene therapy from being delivered to their muscle. This could make them ineligible for a trial. The formation of antibodies is also why redosing, receiving a second dose of gene therapy, is currently not feasible. Once someone has been exposed to a high dose of virus at the time of treatment, the immune system will work to produce antibodies to protect them from future infections. Researchers, clinicians, sponsors, and PPMD all recognize this is one of the biggest challenges to ensuring that all individuals can receive gene therapy and be redosed if needed. Many individuals and groups are working to overcome this challenge. As mentioned before, gene therapy is not limited to delivering microdystrophin. Other transgenes can be delivered as well. These can be replacements for dystrophin, other genetic tools to restore dystrophin, or different genes that provide benefit not directly tied to repairing the muscle membrane.
All right, welcome back. And we're going to start this presentation with Astellas Gene Therapies, who's actually going to talk about one of those non-microdystrophin strategies. So Matthew, you can start whenever you're ready.
Thank you very much, Eric. Appreciate it. And just want to start by also thanking the organizers for inviting us here and providing me with the opportunity to share with you the work that we're doing at Astellas Gene Therapies in Duchenne muscular dystrophy. Next slide, please. Just starting really quickly with my disclaimer slide, acknowledging that yes, I am an employee of Astellas and Astellas Gene Therapies. Next slide, please. So first, just a bit about who we are. Prior to acquisition, we were formerly Audentes Therapeutics, and as of first of this year, we became Astellas Gene Therapies. But really, with this change, not much has actually changed other than the name itself. That at the heart of it, we are very much still the same people with the same mission, and that is primarily to develop AAV-based transformative medicines for rare monogenic disorders with a particular focus on rare neuromuscular disorders. Among our most advanced programs are a pair of clinical stage programs currently being used with patients right now, one in X-linked myotubular myopathy, a second in lysosomal storage disease, Pompe disease. Also, we have a pair of earlier stage programs in an additional set of rare neuromuscular diseases, of course, Duchenne muscular dystrophy, which of course we're here to discuss today, and myotonic dystrophy. With this change from Audentes to Astellas, really the biggest thing that comes as part of this is just the opportunity that this provides us. Now, as part of Astellas, we have access to global resources, which ultimately, if successful, will really aid in our mission of being able to get these therapies to patients and to all the patients in need of these kind of treatments to affect their lives. Next slide, please. So, as was mentioned by Eric and as mentioned in the video, our approach is slightly different than the majority of the gene therapy programs out there, and certainly from the 3 programs that are in the clinic right now. As those programs look to use micro or mini versions of the dystrophin gene, we instead take an alternative approach, but an approach that nonetheless is likely very familiar to this community, and that is exon skipping. So this works on the principle very much the same as the oligonucleotide therapies produced by Sarepta Therapeutics, in the sense that we are introducing a DNA element that can code for a nucleotide that induces skipping of the dystrophin RNA. What's happening here is, in fact, what we're inducing is the removal from the RNA of the part of that sequence that contains the mutation. And therefore, what remains after that is a nearly full-length version of the dystrophin that no longer contains the disease-causing variant. Hopefully then in turn, this provides a therapeutic benefit to the patient by returning a near functional version of dystrophin, a near fully functional version of dystrophin back to the patient, back to that patient's tissues. Next slide, please. Of course, a key partner in our journey in developing these exon skipping gene therapy approaches has been Nationwide Children's Hospital and Dr. Kevin Flanagan. And you'll actually hear a bit more about Dr. Flanagan's work from himself later on in this session. We partnered with him on the development on the clinical trial that is currently being conducted using his gene therapy for the exon 2 duplication version of Duchenne muscular dystrophy. But in developing our programs at Astellas, we've taken a lot of the learnings from this collaboration and generated our own constructs that we're now taking forward through preclinical and hopefully into the clinic as well. One of the most significant changes that we've made in developing our constructs is the shift from AAV9 to AAV8. Part of the reason for this is because of the experience that we have with AAV8. This is the serotype of AAV that we use across all of the programs that I mentioned earlier. And as a result, we have a wealth of preclinical and clinical data and know-how, as well as a lot of manufacturing capabilities, all to support AAV programs utilizing this particular version of AAV. At the end of the day, we think by making this move, it better enables us to successfully and efficiently deliver these therapies into the clinic and ultimately to patients. Next slide, please. So in developing this platform, and what we are trying to do is develop a platform around exon skipping, is the idea that we're building a backbone that can be readily deployed to various different mutations in the dystrophin gene. Because one of the unique factors of this exon skipping approach versus the micro or mini dystrophin gene therapies is that our approach is mutation specific, in the sense that you would only use the exon skipping AAV gene therapy for duplications of exon 2 in patients that have that specific genetic perturbation. Similarly, we're developing programs that skip exon 51 and exon 53 in the dystrophin gene. And again, those are for patients with specific genetic mutations and not for all Duchenne muscular dystrophy patients. As a result, we want to have a flexible platform in which we can quickly change out what we're delivering to be able to continue to deliver different kinds of constructs that skip different exons in the dystrophin gene that will ultimately enable us, hopefully, to deliver a therapeutic that addresses the majority of mutations in the Duchenne muscular dystrophy population and community. Next slide, please. So, as I mentioned before, most of our programs, or all of our Duchenne muscular dystrophy programs, are at the preclinical stage at this point. We have completed both in vitro and in vivo work in rodent species that show that, in fact, we do have constructs that can be efficiently delivered, that they can target muscle and deliver their payload to the tissue that we need it to deliver to, and that once there, they induce this exon skipping behavior that ultimately we hope will deliver the therapeutic benefit to patients. What we have ongoing right now are additional studies, both in rodents and in non-human primates. What these studies are meant to do is to help us both understand the proper dose that we would use once we enter the clinic, that we think will bring the most benefit to patients, as well as understand the safety profile of these molecules to ensure that whatever we deliver, we're doing in a way that protects the patients and their safety. Finally, one of the other activities we have ongoing right now is an assessment of the off-target activities of these exon skipping therapeutics. We're using a combination of bioinformatic and in vitro tools to ensure that when we deliver these sequences to the muscle, that they're only having activity where we want them to have activity, in the sense that they're only going to target the dystrophin gene and not have activities elsewhere in the genome. Next slide. And just to conclude my portion of this session, I wanted to end with a little bit of preclinical data that we've generated around 2 of our programs. The one targeting the exon 2 duplication syndrome, and another one, again, targeting skipping of exon 53. In the first set of data you see here on the left-hand side is data generated in myoblasts generated from a Duchenne muscular dystrophy patient containing this duplication of exon 2. And what you can see in the picture of the gel in the very first lane is a single band at the top of the gel. And this represents the fact that there's multiple copies of this duplication in the patient's cell line. But once we introduce our exon skipping vectors to the cell line, you can see a second band appear. This represents the successful deletion of one of these extra exons from these particular dystrophin mRNAs. And in some patients, you actually see deletion of the extra exon in both of the patient's copies of dystrophin as well. And ultimately, this is how we understand which sequences work best, and ultimately how we optimize and deliver what we think are going to be the constructs that provide the greatest promise of benefit to patients. But ultimately, what we hope this exon skipping will do is result in production of functional dystrophin. And that's what you see in the other side of the slide. This is a picture of myotubes differentiated from myoblasts, again, from a patient with Duchenne muscular dystrophy. And here, what you can see in the images and in the graph below is that we are seeing a dose-dependent increase in dystrophin in these cells with the treatment by our AAV gene therapy. That whenever we introduce these vectors into the cell line, that in fact, it not only is causing the skipping of the exon that contains the mutation, but as a result, you're seeing an increase in dystrophin with an increase in exon skipping. So with that, I will stop and hand it back to Eric and Ryan.
Thank you, Matthew. I just had one follow-up question before we move to the next panelist. You showed in your pipeline exon 53 and 51. I know you're still in early stages, but given that we have approved therapies for exon skipping drugs, ASOs, do you see the potential of these working synergistically at some point or layered on to your therapy?
I think certainly there's potential for that, Ryan, but I think ultimately what we hope is that this will be a standalone in a drug as it relates to those other exon skippers, because certainly what we see as a benefit of the AAV gene therapy version of these is that they will be able to robustly target and deliver these sequences to the muscle. So we'll get sufficient amount of these delivered to the muscle to induce significant exon skippings, and then that there will be a durability to that delivery. So as opposed to the therapies now that require regular injection of therapeutics, that this would be a one-time treatment that could deliver long-term benefit to these patients, because the cells are essentially equipped to make the therapeutic themselves. Great. And will then continue to generate that exon skipping capability within the cell that actually requires the therapeutic.
Great, thank you for that explanation. Okay, we're going to hand it over to Olivier now from REGENXBIO to give his presentation.
Thank you. Hi, everyone. My name is Olivier Danos. I'm the Chief Scientific Officer at REGENXBIO, and I'm very excited and proud to have this opportunity today for introducing to you our new candidate for gene therapy of Duchenne muscular dystrophy, and this is a microdystrophin approach. And I want to thank the organizers, PPMD, for allowing us to present here and giving us this opportunity. My next slide is a slide that I had to show you because we're a public company. Let's go to the next one, please. The next one is a description of our candidate. We are developing an AAV vector, and it's an AAV8 vector that codes for a microdystrophin, and we have our own blend of microdystrophin, and I'll get a bit deeper into explanations about that. So why AAV8 vector? AAV8, you've heard about it from Matthew. It's a vector that's been used in the clinic in the context of neuromuscular diseases. It's been validated, shown to be very efficient in targeting muscle and heart. We have a lot of expertise around AAV8. This is a proprietary vector that we have. It's part of our NAV platform. We have currently 4 different trials in the clinic in ophthalmology using AAV8. So we know how to make it. We know how to also make it at scale for applications that would require a lot of vector, like Duchenne muscular dystrophy. So in the vector, we've designed our own microdystrophin, which is expressed from a tissue-specific promoter called SPC512. This is a promoter which has been used in the field for a long time, never in the clinic yet, but this is a short promoter with a specific activity for skeletal muscle and heart. The dystrophin gene, the microdystrophin gene itself, looks in many ways like other microdystrophins that are being tested in the clinic currently, except that we've added more of the dystrophin domain at the C terminal, and I'll come back on that. So we have a longer microdystrophin that codes for more of the dystrophin. We have also done a number of optimization on this transgene, like reducing the CPG, which we believe potentially minimize immunogenicity initially upon gene transfer. And this is, of course, codon optimized also to maximize protein production. The next slide just gets back on the microdystrophin design. On the left, you have the full-length dystrophin that I won't comment here. The microdystrophin, as you see, retains much of the architecture of the C-terminal. We've added to the current microdystrophin in use about 150 amino acids, and this C-terminal domain, it's the green box marked CT here, this the C-terminal domain allows for binding of essentially syntrophin that in turns will provide a better binding for dystrobrevin, very important part of the DAPC, of the protein complex which is associated to dystrophin. Dystrophin, of course, acting, as you know, not only as a shock absorber, but also as a hub and an organizer of a number of very important membrane proteins. And this is very often achieved through syntrophin, and adding the C-terminal add the power of binding more syntrophin and in turn more partners of dystrophin. One of these important partners, which has been discussed at length in the field, is neural nitric oxide synthase, that's shown represented in red here. And with the C-terminal, we potentially have an enhanced binding of nNOS to our microdystrophin with hopefully a number of important functional consequences. Next slide. So the next slide, this is basically a summary of our preclinical studies in mice. The details are in the pre-recorded presentation that I made here for the meeting, and also at a poster that we have. So if you want to get into it, but basically we've run a number of these studies in MDX mice, injecting young animals, analyzing them at 6, 12, and 26 weeks. We've used increasing doses of our RGX-202. And what we see in a nutshell is a very high level of vector DNA that can be accumulated in the skeletal and heart muscles. That's of course dose dependent, and we have a nice and expected dose response here. Very robust microdystrophin expression in muscle. We actually think that adding the C-terminal may help us there, because we have evidence that stability is increased with this when you add this domain, although this should be fully proven. We of course have evidence of complete DAPC recruitment. We do correct the muscle pathology, if we look at inflammation or degeneration, and we have evidence for reduction of dystrophic lesions through imaging MRI. So, and also functionally, we've been testing strength and gait analysis. So basically, we've tried to make the as complete as possible panel of readouts and endpoints that you can have from the MDX mouse. I'll just show you a few of them. Next slide. This is the MRI analysis, and let's concentrate on the picture panels here. On the top row, you have the analysis at 6 weeks. On the bottom row at 12 weeks, and what you see are pictures, MRI pictures of the muscle. And what we're looking for here are signals of muscle degeneration that appear on the pictures as white zones here. You can quantify these white zones and then therefore quantify the amount of disease. In the healthy control, you see that we don't see any of those. In the MDX, there's a lot of those, and they're indicated by arrows here. And as you increase the dose of RGX-202, reaching dose 3, there's a complete disappearance of these signals, these pathologic signals. The graph bars on the right are just a quantification of that, 2 different quantifications of that. That shows you that at our 2 higher doses, or even at our 3 higher doses, we get a very significant or complete correction of this endpoint. Next slide. This is measuring here at 6 weeks the grip strength or the in vitro forces in the panels which are on the left. In blue, you have the animals treated with 202 that you have to compare to the control in MDX in red, or to the wild type animal in white. In grip strength, we see a pretty full recovery of the ability of the mouse to perform in the test, and in vitro force we get significant amelioration also in this assay. Gait analysis was something that we found interesting to measure. There are actually good ways to test that in these animals, and there's a scale which has been developed to quantify the ability of the animal to walk in a proper way, basically. As you see, the score is pretty high for MDX in red here, and lower for the wild type animals. And again, in a dose-dependent way, we see a correction here in this assay at 12 weeks. Next slide. So just where we are quickly, we've optimized microdystrophin. We've actually tested a number of them. We came up with the one I'm showing you today, RGX-202, because we think it has the best activity we've seen. We've obtained preclinical proof of concept, and then we are in the final stages of putting together an IND application. I've shown you data in mice. We're still putting together the final reports for the long-term data at 26 weeks. We have a safety and tox study in non-human primates as well, testing our doses, and this will also go in our IND submission. Very importantly, and I insist on that, we have commercial scale capacity now in-house at REGENXBIO for production of AAV8 vector, and in particular this vector. And we are also in the final stages of designing our clinical study. So all this will lead us to the IND, which we've indicated will submit around mid-year this year. Importantly, we've been engaged with the community. We think it's highly important and of central importance for developing the trial and understanding the unmet need here. So thank you again for the invitation, and I'll stop here.
Great, thank you, Olivier. Just one quick question. You mentioned in your presentation codon optimization in the description of your microdystrophin. I mean, this is probably something new to families. I think it's so important to explain in the easiest terms possible. Could you talk about this codon optimization piece?
Yes. So this is something that pretty much now everyone designing a gene therapy vector, whatever the application, where you have a protein coding sequence, everybody does this codon optimization. It is a way to whereby you choose for each amino acid your DNA, you have different possibilities for codons. And human cells use a particular codon in general, which is not necessarily the same codon as bacteria or even mice sometimes, right? So if you're careful, you can make sure that you use the right codon. You always have an opportunity, and there are now software doing that, and so it's basically an optimization of the way your protein is encoded by the cDNA. Again, there are different possibilities for that, so you have to choose between actually a lot of different possible versions.
Wonderful.
And usually it results in improved protein production from a given DNA sequence.
Wonderful. Thank you so much for that explanation. Okay, I'm going to hand it now to Kevin Flanagan from Nationwide Children's Hospital for the final presentation in this segment.
Thank you.
Thank you very much for the invitation to speak. And it's a pleasure to follow Matt and Olivier. I'm going to discuss today our work, a little bit of our preclinical work, and some early results from a first-in-human clinical trial of treating duplications of exon 2 using the version of the exon skipping vector we developed here, which is in AAV9, and discuss a little bit of that data and safety related to it. So next slide, please. I should have included a disclosure. I apologize, I don't have a disclosure slide. I will acknowledge that this is actually patented, and the patent is licensed to Astellas, who has supported work in our laboratory as well. We've presented work at the PPMD. In fact, we presented much of this work over the years using this idea of viral-based exon skipping. And just to emphasize a couple of points that Matt already made, what this really consists of is a stably produced RNA that is not translated into a protein, the U7 small nuclear RNA, particularly is stabilized in the presence of SM protein and another cellular protein. And this means that you create RNA that can be targeted to a given exon sequence, and it is continually produced in cells. It interferes with the assembly of the spliceosome. Not being translated into protein, we're not worried in the same way about immunity to what we've delivered. And in answer to one of the earlier questions from Ryan, we know that it is quite stably transduced stably transcribed for a long time, meaning that it is really, we're talking about one-time therapies, or most long-term therapies that might need redosing over time, rather than the weekly dosing of for example, the current commercial PMOs. Next slide, please. The version we have actually is directed toward exon 2. We picked exon 2 because of a variety of reasons. First, it's the most common exon duplicated in the gene, in the DMD gene. And second, because we have a large safety margin. If we skip both copies of exon 2, we still can make use of a highly functional protein that's translated downstream, beginning with translation in exon 6. Due to what's called an internal ribosome entry site. But it just seemed a very good exon to start with for those two reasons. And we optimized sequences that targeted either the splice acceptor or donor site. And the vector that we've used in our development program so far has been an AAV9 that contains 4 copies, 2 copies each targeting the splice donor and the acceptor site. Next slide, please. So we have shown that with we can look at this in a couple of different ways. This is really recently published data, although it goes back a way. It was involved in our IND filing. This is 12 weeks post-injection of 8-week-old mice that are injected with the vector. And again, on the left side in panel A, you see the bands that Matt talked about before. In fact, you see as we go up in dose from column to column, we can see a top band that represents the duplicated transcript. We can see a middle band that consists of the wild type transcript with 1 copy, or we can see the lowest band that consists of no copies of exon 2. So again, we know that these 2 transcripts that either contains just a single copy or no copies at all, both of those are therapeutic transcripts. And in the B side, we've represented this here. Any of the shaded columns is the total therapeutic transcript that we see, both wild type or the delta 2, here we call it delta 2 transcript. And there's a dose-related response as we increase. So we know skipping occurs. What was necessary then, if you advance one more time, this is actually a description then of the protein that we see expressed at the same time point. This is a representation of protein that you may not have seen before. We have a paper just going off for submission now describing a new automated methodology that we have that we're very happy with to automatically count dystrophin positive fibers, which you see in the top in panel A. That's the red staining on each of these. And then below that, a heat map of a processed image that actually tells us for those positive fibers how much of the circumference of the fiber is in fact positive. And then in the bottom model, we have what we show to be intensity, overall intensity of this. What we can see here, you can see it in the graph, you can see it by eye, that there is clearly a dose-related response to this. And a 2.9 e to the 13th actually corresponds to where we are with the starting dose in the trial of 3 e to the 13th, or 3 times 10 to the 13th vector genomes per kilogram. We can advance, please, if you would. We actually have an extensive safety package using this, and we've just published in May showing data in non-human primates at these dose ranges that there's no significant toxicity associated with it. And in fact, just in PubMed now is data from a study where we looked extensively for the off-target effects that Matt mentioned earlier, in which case we're looking to see we want to change splicing in exon 2. Do we have some random or potentially detrimental splicing effects? And actually, we took a methodology that was a non-biased approach to look at all exon-exon junctions throughout the expressed portions of the genes in mice, and we can see no evidence of significant off-target effects. So we think the specificity is quite good. We could advance one more slide. So I'm going to share some really early data. This has been presented at the American Society of Gene and Cell Therapy by my colleague, Megan Waldrop, because of conflict management purposes, and among other things, Megan is directing the trial here. And this is results from 2 patients who've received this therapy who carry duplications of the gene. If you can go to the next slide, please. Both subjects began a prednisone regimen of 1 milligram per kilogram per day prior to treatment and received this over 90 minutes. There's 2 subjects, one treated at 8.9 years, one treated at 13.7 years. Both of them had some adverse events related to abdominal pain and nausea and vomiting that we're actually familiar with in systemic AAV therapies, AAV trials, and AAV therapeutics actually in the case, for example, of Zolgensma, but no other significant side effects related to it. If you could go one more slide. This is on the left are transaminases, AST and ALT, which again we do monitor. We want to point out to parents, and I want to take this opportunity, as we often do, to remind parents that these transaminases are often called liver function tests. But in fact, these transaminases are released by muscle as well. And we know that they track and correlate with serum CK measures. But we do follow them as well for markers of liver injury after systemic AAV delivery. And we see one of the 2 patients had a bump in his ALT in the first 2 months of his treatment, but in fact we saw no sustained or really worrisome numbers, and these did not correlate with changes in another enzyme I haven't showed you here called GGT, which is specific to the liver. More striking on the right is the serum CK levels. So creatine kinase dropped really precipitously in the patients in whom got this. At days 45, they were, in fact, in levels we just don't see in boys with Duchenne dystrophy, as you can see from the graph. These are ad lib activities allowing the boys to do whatever they wanted. Even at the most recent follow-up for these, they stayed quite diminished, 35% to 28% of baseline. If you could show the next slide. I'm just going to show you some Western results. These are not the final results. These are preliminary results. To explain that, these were done and part of the development in a colleague's lab who's doing the blinded staining of all samples. So, but in these preliminary results, the formal blinded one will be done after the final biopsy is completed on the second patient. But we do see measurable dystrophin. You can see it best in subject 1. These levels are not exceedingly high. They're 5.8%, as you can see in subject 1, about 1.5% in subject 2 at 3 months of time. I will note that we think this is the first example of restoration of full length of dystrophin in a gene therapy for Duchenne dystrophy. I'll also note that we and others have published that levels in this range actually clearly can ameliorate disease. Now, we're not satisfied with this, and we want to increase the dose and plan on future studies that look at dose ranging escalation, particularly with a new vector being generated by Astellas. But we do know that these are sufficient with ameliorating disease in patients and in boys who express some degree of dystrophin because of their inherent mutation. So next slide. So I'll just point out that we can draw some conclusions. This is interest in administration has been well tolerated. Creatine kinase levels showed a marked improvement. Actually, functional data I didn't show, but there was really improvement actually in the 100-meter walk time in subject 1 and stability in subject 2. As well as stability in the North Star ambulatory assessment values in one subject, improvement in the other one. We don't want to draw too many conclusions from this N of 2 study regarding functionality, though, at this present. We do see an increase in dystrophin expression, and again, it's striking for us, it's full-length dystrophin expression, but I think this clearly supports further clinical investigation. Primarily, we want to concentrate on younger patients, and the data, I would say, suggests supports the idea of higher dosing. So we could advance one more slide. I just want to acknowledge Dr. Waldrop, who's leading the trial, and Dr. Wen and Dr. Sheffield, who helped develop this therapy, and our colleagues at Medical College of Wisconsin, Mike Lawler, in particular, who's leading the blinded analysis for Westerns, and note that the work's been supported by the synthesis of the vector that we used was supported by the Bohawks Foundation, and we had very early support in the project from Cure Duchenne. So with that, I'll close. I'm happy to take any questions.
Great, thank you so much, Kevin. I'll do one question before we open up the panel. You now have dosed an older patient, higher weight. As trials are moving forward and you're thinking of dosing more older patients, heavier patients, do you have concern about the total viral load for these patients that they'll be receiving?
We do. That's always a concern. Although we're at a relatively low 3 to the 13th, even with that size of a boy, the total load is still not excessive in comparison to some other therapies that have been given, but it always, of course, will be a concern. He is one of the larger people, given his weight, that we've treated with systemic delivery.
Great, good progress. Eric's going to open up the panel for discussion now.
Yeah, and so again, if you have questions, please submit them to that comment box below. And so this first one, actually, I think everyone can touch on, but especially with Astellas and Regenex. It'd be great to touch on some of the lessons that you're learning from other companies as you're entering the space. You also have experience, yourselves and with other diseases. We talked a little bit about this in our other gene therapy discussion, but rather than using something like age for inclusion criteria, are you thinking about things like using function or weight rather than just simply a 4 to 7 designation? And Olivia, maybe I'll start with you and get your thoughts on that.
Yes, thank you. That's a very good point. And yes, we do think about that. Age is some kind of an arbitrary point, arbitrary thing to do when you look at the diversity of the disease and patient. So it would be much more appropriate if we could have some kind of test where we would stratify our patient with. But age is been criticized and it's been discussed a lot in the field, and but this is the standard and this is what everybody is doing. So you start with that, and I think not only us, the field is trying to improve that. I'm hopeful that this will happen. But yes, that's absolutely a good point.
Great, thanks. Yeah, go ahead, Kevin.
Sorry, I mean, I just want to, as a trialist, I want to inject one note of caution about that, though, particularly with the dystrophinopathies, where there is the possibility for enrollment picking those patients who are doing better, for example, and particularly because there are things that are called dystrophin mutations. There are children diagnosed and carrying the diagnosis of dystrophin who do appear at the, after 10 years after diagnosis, to have a somewhat different clinical course. Often in our work that we've done, it's often due to very low level expression, measurable, but low level expression of dystrophin. So there is a caution to be said if function alone is a determinant. A 7-year-old boy with Duchenne who falls at a level, there might be another boy at 10 with the same degree of function. We have to be cautious. They might represent different baselines amount of dystrophin.
Right.
And we want to enroll. We don't want investigators picking boys who are the best function to start. It'll confuse the readout for these studies, I think. So I appreciate the importance of this, but I think there's a caution to it as well. It has to be thought out very carefully.
No, we appreciate that response. I think it's really helpful for the community as we kind of think about this and have these discussions. And Matt, if you have anything to add, I would love to hear it as well.
Yeah, not too much material to add to this. I mean, given where we're at with our programs being preclinical, we haven't actually set what our inclusion and exclusion criteria will be, but obviously we're already thinking very deeply about subjects like this. And I think, as Kevin, nicely outlined, ultimately there will be multiple factors that fall into how we think about enrolling patients. I think Olivier is right. Function is going to be part of how we think about this beyond age. Certainly weight and dose are going to be part of this. Other factors such as existing neutralizing antibodies, which again, I know was touched on in that video ahead of time, all are part of the calculus that we ultimately do in trying to identify the patients we think are going to be get the most benefit out of these early trials and could possibly give us the best understanding of the therapeutic benefit of our molecules in these trials.
Yeah, great, thank you. And actually, on that point, because we have this question coming in, and I think it's a perfect time to go to it, is on neutralizing antibodies. You have experience with other diseases. Are there ways that Astellas and Regenxbio have been looking at this and potentially strategies they're thinking about for Duchenne or exploring, in a preclinical setting for how to get around this challenge? And Matt, I'm just going to throw that back at you.
Sure, certainly. No, this has been very much on our minds because we do recognize that this is a huge barrier for patients actually being able to access these therapies if and when they ultimately get approved. And that right now, unless we find solutions to it, there are some proportion of the patient population that simply may be ineligible. So certainly, we and many other people in the field have been looking at this. To that end, we don't have anything that we're prepared that's sort of mature enough for us to talk about in this form. But I'll say that in the space, if you look at what's happening, there's work by others who have used, for instance, enzymes that are able to degrade neutralizing antibodies. And that could potentially be used as a pre-treatment before the gene therapy as a way of removing that barrier. Plasmapheresis is another technique that has been proposed as a way of being able to clear these. So again, there's a number of investigational avenues that people are pursuing because I think we do recognize that this is something that we want to ultimately be able to address.
Great. Thank you. Olivier, if you could comment on if there's anything that Regenx is looking at, early stage as well.
Yeah, I think Matt described the approaches that the field is looking into, and we are, trying to eliminate the existing antibody is something that's an active area of research. But I'm saying it's research. This is not something that has been translated in clinical trials yet. It will take a little while, but it will get there, right? I think for the current trials, nobody really wants to take chances, and for the safety of patients and for our ability to read out correctly, we will include patients that are negative or have very, very low levels of antibodies to the corresponding vector, whether it's AV9, AV8, or others. Then the name of the game will be to be able to include others eventually by adding this kind of interventions, immunomodulations. I mean, all kinds of things that are being looked at, but nothing today is absolutely ready for prime time. But there are a lot of there are different ways you can think of solving the problem, and they're actively looked into.
Great, thank you. And Kevin, I'm going to ask you this because you might not be as tied to timelines and can share your thoughts. Is this something you think is going to be explored more after we have approvals of some of these gene therapy products, or do you see it kind of evolving alongside kind of our current clinical trials?
I think it's evolving alongside. As Olivier said, I think all of these early trials have concentrated just on safety of these products themselves. As we have more data for these products and the class of products, I know not only Astellas who we've worked with, but lots of other companies we've talked to. It's really there's a deep understanding of the need in the community for this. There's obviously a commercial necessity for it. So a lot of groups are moving forward. There are an increasing number of trials in the design, or even now early phases or trial phases in other diseases where there's talk or implementation of preconditioning regimens for patients before they receive the AAV. And I think we're going to see that more commonly, and that will translate as well to preconditioning for patients who have particularly existing environmental exposures to AAV. So it's be my prediction.
Great, thank you.
It feels like we're learning as we go. In some ways that can be scary, but it's moving fast at the same time, so it's good for the progress. So this is for all of you. General question, we've been talking about MRI throughout the conference, both cardiac and muscle for fat fraction. Are there plans and thoughts about utilizing these within your studies? And I could start with Matthew.
Yeah, I'll circle back to what I mentioned before, just because of the phase that we currently sit in. It's all sort of plans right now, but certainly we're aware of the progress that's being made in this area and the potential benefit of really being able to, in much more real time and in a non-invasive way, understand the benefit that is being seen, if there is one to be seen. And so certainly this is something that we continue to think about and explore as we put together what our clinical trial would look like.
Yeah, I think we need to be thoughtful about how are we measuring the impact on the heart. I don't think we really know yet, and it's something that needs to be looked at. Olivier?
Yeah, I think we're very excited about the possibility to use MRI as an endpoint in our trial. This is something that we're still looking into, but this is not just a nice idea anymore. This is something where groups have worked a lot around that. There's a lot of data. There's a huge database and very sophisticated means of investigations that are out there that are clinically translatable. We believe that this is almost ready, and as soon as we can, we'll introduce that in our clinical studies.
Okay, and Kevin, do you want to comment?
Well, I'm going to sound always like the wet blanket, I guess, in some ways. I think it's value. We have it in trials of this and GAL-GT2 and other ones. But and there is, as Olivier points out, really great data coming out of different groups about MRI as a biomarker and progression. I think there's still some gaps in the real prognostic implication of MRI changes over time. There's still variability in patients among them. Yeah. I think the more patients we do, we get more information. But the correlation, say specifically for a dystrophin replacement therapy, such as the U7 therapies, let's say, I don't think we can necessarily. We still have this issue of showing that there is in fact specific replacement of the dystrophin at the membrane that we want. And although you could argue that, well, if MRI predicts function and MRI shows change, we don't really care how much dystrophin is there. I think we're still obligated to show that. In other words, I don't think we're going to use. What I'm really trying to say is I don't think we're going to be able to use MRI to get away from using biopsies in studies like this, which is a question parents often ask me about.
Absolutely.
Yeah.
Particularly
I think
where we are in the field, I think it's critical. We understand that. Less.
I'm sorry, Olivier was going to say something too. I didn't want to. You had something to add, Olivier. I didn't.
No. I mean, I just wanted to agree with you. That is, this is not going to be the magic endpoint that will replace others. And again, this is the solution is going to be with a variety of endpoints and a totality of evidence on different endpoints. That's for sure. And it's great that you've made these remarks. I mean, even. I would be cautious with being over enthusiastic on a single endpoint, whatever it is, whether it's a functional one, whether it's a biochemical one. But the good news is that we're starting to have a whole panel of things that are very solid and very serious, and that's great.
Great. Well, I want to thank the panelists and for all you're doing. We really, really appreciate it.
Thanks for having us.
Thank you.
Thank you.
Okay. Bye, everyone.
So now we're going to actually spotlight a few PPMD programs and funding programs and be right back.
The Duchenne Registry is an international registry for people who have Duchenne and Becker muscular dystrophy. Women who are carriers are also invited to join. A registry is a place to collect information or data about people who have a specific disease or condition. By sharing your data through the Duchenne Registry, you're joining a global effort to better understand Duchenne and Becker. Doctors and researchers use this data to improve care and develop better treatments. None of these people can do it alone. We need you. Join the Duchenne Registry through our new user-friendly smartphone app. Learn more about the app at DuchenneRegistry.org, where you can watch how-to videos to help you get started. Once in the app, you'll answer some surveys. Based on your answers, we can send emails about research studies you may qualify for, since many researchers use the registry for recruitment. Your privacy is important to us. Everything is stored securely. We never share your name or contact information. If you have registry questions, you can talk to one of our genetic counselors on the phone or by email. In addition to helping you with the registry, they can interpret genetic test reports and review the impact on your entire family. If anyone in your family needs genetic testing, our genetic counselors can coordinate free testing with your local doctor through our Decode Duchenne program. They can also discuss research opportunities and potential therapies. You have the power to make a difference. Join the Duchenne Registry and join the fight to end Duchenne.
Hello, I'm Ann Conley. I'm the Division Chief of Child Neurology at Nationwide Children's Hospital, and I'm excited to present to you a brief summary of the grant that was awarded to me to see if once-weekly corticosteroids are safe and effective in infants and young boys with Duchenne muscular dystrophy. This study is being done in collaboration with 3 sites, Dr. Nancy Kuntz in Chicago, Dr. Tien in Cincinnati, and Dr. Ayana Cohn at UT Southwestern. One major reason we believe that a smaller dose may be effective is recent mouse and work with Duchenne boys from Dr. Quattrocelli in Dr. Beth McNally's laboratory period. This work shows that weekly corticosteroid exposure blunts the metabolic stress that happens in daily treated boys. Despite the lower dose, this dose was effective both in boys and in mice, and we hope that it will be equally effective in the infants. We hope to enroll 12 boys in this study, which we hope to complete within 2 years. To fully flesh out the comparative efficacy, it will take additional boys, but this study funded by PPMD will go a long way in getting us started. Thanks very much for listening.
Thanks, Anne. Such an important study. And again, with the Duchenne registry, if you join the registry or you fill out all your surveys by the end of this month, you'll be put into a raffle for a smartphone, Android or iPhone. So please make sure to do that. We're actually going to take a break and come back at 3 p.m. Eastern, and we're going to have new data from Reveragen and Eric Hoffman. We will see you at 3. Welcome back. We will now be joined by Dr. Eric Hoffman to present recently reported updates from the phase 2 vamorolone trial. Eric, take it away.
Thanks. Good afternoon. Can I have the next slide, please? And this is a disclaimer for Synthera. Next slide. So what I'll be talking about is a drug program we've worked on for the last 10, 12 years. The goal is to first understand why corticosteroids work so well, but as well to understand what causes their safety concerns or side effects. And we know that, and you've heard through this conference by a number of talks, that corticosteroids, both deflazacort and prednisone, improve strength, prolong ambulation, arm function, delays ventilator use, and delays scoliosis. But with that efficacy comes safety concerns. So weight gain, bone fragility, growth stunting, cushingoids, suppressed immunity, and mood and behavior changes. Now, some of these safety concerns themselves can exacerbate the disease. For example, I'm sure you've all heard of DMD patients who break a leg and that they lose ambulation because the leg breaks. And that bone fragility is a safety concern, a side effect of corticosteroids. So the goal of this program is to try to understand both efficacy and safety and separate them. See if we could come up with a drug that retained the efficacy but could lose some of the side effects or all. Next slide. So what we landed on, it was John McCall, Kanabun Nagaraju, and myself, was a drug called vamorolone, previously called BBP-15. And the key is sort of in the northeast there, that H, where all other corticosteroids, there's 33 other approved corticosteroids like deflazacort and prednisone in clinical use worldwide. Fastest Nobel Prize ever awarded in 1950 for these corticosteroids, which are amazing drugs. We just changed that, they all have an O or oxygen there, and we changed it to a hydrogen. And what that does is we know now through lots of work with both academic labs and our own labs, is that changes how this drug interacts with its receptor, the glucocorticoid receptor, and it changes the downstream properties. And what we're seeing is this separation of efficacy from safety concerns. So key difference is that vamorolone is not a corticosteroid. Is first, it lacks that oxygen, unlike all the other corticosteroids. But it also reduces its effect on genes. So it doesn't turn on the genome, all the genes. Corticosteroids turn on about 10% of the genome by many published papers. We don't do much of that, and that seems more associated with safety concerns. You've also heard in this conference that eplerenone, spironolactone, are heart healthy. They're cardiac drugs, and you've heard that also they may improve skeletal muscle function. Well, they're mineralocorticoid and receptor antagonists. They bind to a different receptor than prednisone and deflazacort. We do the opposite to that. So we're an antagonist like eplerenone. So we have this new activity that makes us like eplerenone, spironolactone, at least in mouse studies. There's also HSD enzymes. These are 11 beta HSDs, which are in all our cells and tissue, and they carefully regulate whether prednisone or prednisolone is active or inactive. The same with deflazacort and cortisol that woke you up in the morning. We're not a substrate for those. So because they work on that oxygen and we don't have it. So those HSD enzymes are different. So the goal is then to see if we have in fact separated efficacy from safety concerns. Next slide. So we did our initial trials in Duchenne muscular dystrophy. I mean, this drug, if it is a replacement for corticosteroids, may be broadly used in arthritis and others, but this was all done by Duchenne scientists together with Duchenne foundations from the beginning. So we just focused on Duchenne. And we did a large first in-patient study. Many drugs you have to go in adults first, but here we went straight to Duchenne kids after a phase 1 study in adult volunteers, but in pediatric patients, Duchenne patients. And we did a large first in-patient study of 48 children, Duchenne children, 4 to 7 years of age that hadn't been treated with steroids. And we did first a 2-week on, 2-week off, which is a PK safety study. And we did a very broad dose range. We did a 24-fold dose range from 0.25 mg per kg, about a third of prednisone dose, up past 10 times prednisone dose. So a lot of different doses to really get a feeling for that safety efficacy balance. Then the patients were rolled onto a 6-month extension study at those same broad doses. And what we found, as you can see off to the right, is the 2 higher doses showed improvement on many of the outcomes, the motor outcomes, whereas the 2 lower doses didn't so much. So we were shooting for the 2 and 6 mg per kg going forward. So then those patients liked, and the families wanted to stay on vamorolone. So it was a 2-year extension study. And 41 patients finished the whole 2 years, and they all dose escalated to the 2 and 6 mg per kg. Now that was recently published in, by Eddie Smith back in 2020. He was one of the speakers earlier in this meeting last fall, and that showed that the kids continued to improve past 6 months through a year and a half on these outcome measures. And the improvement seems similar to corticosteroids. And there we started seeing better safety profiles. We didn't see the stunting of growth that you saw with corticosteroids. The bone biomarkers looked better. There were less physician-reported safety concerns like cushingoid or mood disturbances, but it was an open label study. And you always have to be hesitant about making any conclusions from an open label study where you don't have head-to-head comparisons against placebo or prednisone. Those patients have about a year ago finished a full 2.5 years of treatment. And Jean Ma in Calgary is writing up that data now and should submit a paper. But again, improvement relative to baseline, and again, loss of stunting of growth and seams of bone morbidities in an open label study. So next slide. We wanted to go on to your sort of gold standard, placebo-controlled, double-blind registration study. The goal is, this is the type of trial that FDA and EMA and all regulators like to see in order to get the drug labeled or approved. So this was 121 children with DMD, boys, same 4 to 7-year-old, steroid naive, and they were enrolled in 11 countries at 33 sites. Many of the sites were the Synergy site groups. So you've heard almost many talks throughout this conference of the CINRG. That's a network of collaborating academic clinical trial sites that's worked together for 20 years. And the Synergy group has also led these vamorolone trials. But we included a bunch of other sites as well in countries that like Greece and some others that weren't so experienced with clinical trials to be inclusive. So the patients were randomized into 1 of 4 doses, 1 of 4 groups. You see there in that treatment period 1, either the lower dose of vamorolone 2.0, the higher dose 6.0, so that's about 3 times prednisone to almost 10 times prednisone. And then we had a prednisone arm as well, so a blinded comparison. And then we had placebo, so 25% on each. So that's the data I'll present today, which is this period 1. Now those patients, after they finished period 1, went on to all onto vamorolone, including the placebo and the prednisone crossed over to vamorolone. And those patients are just finishing up, so I won't show that today. I should mention that all the patients, both in the older trial and this trial, that finished the trials can go on to expanded access or compassionate use, and the vast majority have asked to do that. And so those programs are active in all 11 countries. Next slide. Now what you do is you have to, in a double-blind gold standard trial like this, set up your primary endpoint, your secondary endpoints, and your exploratory endpoints. And because we were doing 2 doses and also prednisone, we had to line these up and all agree with FDA and EMA how they're lined up. And you're only allowed to start at the top and see if you win on the first hand of cards before you go into the next one, before you go to the next, before the next. So our first primary endpoint was the higher dose of vamorolone versus placebo, because we wanted to see efficacy versus placebo. And that was the primary outcome. And then if we win on that, we went to the secondary outcomes, which were then the lower dose versus placebo, 6-minute walk at both doses versus placebo, time to run walk, and then exploratory. We didn't line them up, but time to climb 4 stairs and North Star. And you've heard of many of these outcomes throughout the meeting. So now I'll show the results of this trial. Next slide. First, the randomization looked very nice. So this was all done by somebody overseas and everybody's double blind. So the patients were all randomized to these 4 groups, but the groups look very similar. Next slide. So this was our primary outcome. So this was the higher dose of vamorolone in red versus placebo in gray. And there were about 28 kids per group. And you see the placebos are declining a bit in speed. So this is velocity, how quick they are. Whereas you see improvement with the 6 mg per kg. So this was highly statistically significant, 0.002. As you see there, you have to get less than 0.05. And so we hit our primary outcome. Next slide. If you just change that into seconds, how many seconds it takes the boy from lying on their back to standing up straight, you see that, again, the placebo boys are taking about ½ a second longer on average, whereas the boys on vamorolone at the high dose are speeding up. So they're taking about a second less time. So about a second and a half or 1.7 seconds difference over 24 weeks. And this is similar to what we had seen in the previous open label study. Next slide. If you superimpose now the prednisone group on top of that, you see that vamorolone is performing very similar to prednisone. Now here I want to make an important point. Is prednisone in this study was 0.75 mg per kg per day, which is the recommended dose by the AAN, American Academy of Neurology, and published papers, standard of care. But an important thing is that the majority of patients don't get those doses that are recommended. You heard that from Craig McDonald in the placebo arms of published studies, where in fact they're getting about 75% of the recommended dose, both for prednisone and for deflazacort. That was also seen in the Synergy natural history study of 440 kids, about 25% reduction in the recommended dose. It's because everybody's trying to balance safety concerns and efficacy. So here, hopefully, if we can reduce the safety concerns, there'll be more flexibility with dosing and not so much of that struggle. So that it won't be underdosed necessarily. Next slide. Well, let's look at all the other outcomes. So what you see is we hit basically everything. So you see the primary, secondary, and exploratory all showed improvements relative to placebo at both the low dose and the high dose. And you see those p-values are all of them less than 0.05 by a lot, except the one there, time to run walk velocity at the lower dose, sort of just missed. So this is very compelling evidence that vamorolone improves Duchenne kids and the p-value says quite impressively versus placebo, and all those were similar to prednisone effect. One thing I should note at this point is that you hear often in this young age of the honeymoon period. In fact, many trials, even I think the Sarepta gene therapy trial, showed improvement of these kids even without the gene therapy in this age range, the younger age range. Well, I think a lot of that's from steroids, because steroids, we know, as we showed in this trial as well, do a dramatic improvement of Duchenne boys. So from our trial, you see that if you don't give them steroids, they're in fact declining, not a lot, but declining over the 24 weeks. Next slide. So let's look at safety. Again, we're trying to separate efficacy and safety. Now, one thing you can do is just ask the physicians and the families to count up adverse events. And so we see hints that prednisone has higher adverse events than the 2 vamorolone doses, but these are serious adverse events were very low. So there was only one serious treatment emergent adverse event leading to discontinuation of the study, and that was in the prednisone group. Next slide. Now, if we dig into adverse events a bit further and look at adverse events that are associated with corticosteroid use, and that's this pre-specified safety analysis off to the right, you see that the vamorolone doses do seem to show less of these corticosteroid-like safety concerns reported by families and physicians, and that's significant. Next slide. So one of the things we had seen, as I mentioned earlier, in the 2.5-year study is that all the kids on vamorolone grew normally. In fact, they grew a little faster. There seemed to be some catch-up growth than their peers. That's in marked contrast to all corticosteroids when given to a child at chronic doses stunt growth. So corticosteroids are well known to shut down bone remodeling and just halt growth, and that's part of probably making the bones more brittle as well. And so what we saw was we confirmed even in 24 weeks, this short time period, 6 months, that in fact, vamorolone kids at the higher doses were actually catching up in growth. They're going, that's off to the left there, they're actually showing a mean of 4 percentile change. So they're catching up to their peers. Whereas we see the stunting of growth with prednisone that you also see with deflazacort and all corticosteroids. And off to the right is that 2.5-year treatment. And you see that normal growth is maintained over 2.5 years. Next slide. Now, this was new data of a head-to-head looking at bone biomarkers. So bone biomarkers, or bone remodeling biomarkers, have been used in nearly 500 clinical trials in all sorts of disorders and in 4,000 publications, because you can take a blood test and sort of look at these 3 protein biomarkers and see if bone is remodeling or not. There's sort of a surrogate marker for bone remodeling. Is bone growing or remodeling or shrinking? So what you see in the black bars is prednisone in this trial, this blinded trial, really shut down bone remodeling. Again, that's very consistent with what corticosteroids do to bone in anybody, adult or child. But you see in the placebo, the gray, they didn't do much change. But with vamorolone, they actually increased in a number of instances. So that we have separated the bone morbidities. So we're not doing the changes in bone biomarkers. We're not seeing the changes in growth that you typically see with corticosteroids. Next slide. Weight gain is another concern. Now, with weight gain, it's not everyone on deflazacort or prednisone that gains a lot of weight, but some kids seem to be sensitive to it. So we basically see the same thing with vamorolone. There's a subset of kids that seem to put on weight, and that weight gain is similar to prednisone, or a similar proportion of kids and similar amount of weight gain as prednisone. What we see in practice in the long-term safety study is that at the higher dose, if some physicians saw some weight gains, they can go to the lower dose, and that in some instances seem to help that. So again, you see even the lower dose group, there seems to be less weight gain than you see with prednisone, though that's not statistically significant. So it's about 5 to 26% of kids on vamorolone started to see this weight gain. Next slide. So in summary, vamorolone is being investigated as a first-in-class dissociative steroid with lower incidence of corticosteroid-associated adverse events. This double-blind 24-week study showed statistically significant and clinically relevant improvements across all the motor outcome measures we looked at across a broad dose range. So it's both a threefold dose range that hit in both efficacy and safety. Vamorolone did not adversely impact growth trajectory or biomarkers of bone health. Treatment was well tolerated with fewer physician-reported adverse events typically seen with corticosteroids at 24 weeks in a head-to-head with prednisone dose-dependent manner. Vamorolone is the first dissociative steroid. So to our knowledge, this is the first time that a drug has been successful in separating the efficacy from safety concerns of corticosteroids. And surprisingly, we found this over quite a broad dose range, a threefold dose range. And that will allow physicians to tailor treatment to the individual without losing efficacy, because we see significant efficacy at both dose ranges, both the low and high. Next slide. First, I'd like to thank all the foundations. PPMD has been a great supporter of this program. I should note that many of these foundation supports are under a venture philanthropy model. So when vamorolone hopefully gets sold in the future, they'll get paid back plus 400%. So this is a shared risk, shared benefit business model, which we hope will bring funding back to the foundations. Also want to particularly call out a couple of these funders. The NIH NCATS gave us a trend grant, Therapeutics for Rare and Neglected Disease, and really did a huge amount in helping with the early drug development of vamorolone. And then the clinical trials were supported by NIH NINDS, who is a major supporter of Duchenne research, and recently with a commercial registration pilot. And then also the European Commission, European Community provided a large grant to Newcastle University in support of the clinical trials for this. Next slide. And this is my last slide, just to thank the families first and foremost. We know this is a heavy lift to join these trials. We've gotten some feedback from families, and we hope our trials we try to keep visits down, and then COVID hit. And we're so grateful to the families and all the sites and the staff to doing the quick pivot during COVID. So we didn't lose a single patient in this trial while it was done during COVID. We didn't lose a single primary outcome because we worked with the families to do the time to stand using videos. And a shout out to Newcastle and Meredith James, who was critical in doing this very rapid COVID pivot to finish this trial in the time of COVID. And these are the sites and the physicians that worked with us, and the wonderful physicians at each of these academic clinical trial sites. And the study chairs, Michaela Guglieri at Newcastle and Paula Clemens at Pittsburgh. The Synergy Group, CINRG, spun off a coordinating center, which is TRINS LLC, and they ran these trials and did a wonderful job. You don't get p-values like that unless the trials run well. Camden Associates were medical and clinical monitors, and really we called them the adults in the room. They did a phenomenal job overseeing these studies. Summit Analytical for Statistics, and the operations, Jesse Damsker, who's a master ceremonies, made sure the drug got to the right place and worked with all the sites. Thank you very much. Happy to take questions.
Thank you, Eric. I think we have time for just a few questions, quick questions. So we're, of course, tracking all of the sort of payer activity happening in the first approved therapies with Exondys, Amondys, Viltepso, et cetera. Those payer policies are requiring a 6-month steroid, corticosteroid use in patients to be used along with those therapies. Is there any concern? So you've talked about vamorolone being a dissociative steroid. Do you think payers are going to allow for that as the steroid to be used with those exon skipping therapies? Sorry.
So your question is, will vamorolone be an add-on to exon skipping and other therapies?
Yes. Well, payer policies, from what we're seeing in the access space, is the payers are requiring that patients are on steroids if they're going to get it covered for exon skipping therapies. So I was just curious of your take on what payers may think about if a patient, if this is approved, the patient is on vamorolone, will that count to be included as an add-on with an exon skipping therapy?
Well, the intent of vamorolone is to replace corticosteroid use. You'd never use corticosteroids and vamorolone at the same time. Yeah. I mean, you can transition probably from one to the other, like even in our trial, patients transition from corticosteroids, prednisone, to vamorolone. But we would be a different standard of care. So, I mean, we'd be, and you'd think that let's assume it's approved. I mean, the NDA will probably go in next year. Our feeling is that this data I showed you is sufficient for approval, but that obviously you have to go to the regulators and see if they agree. But once it is offered and labeled for Duchenne, then it all is that discussion with payers that you see with all these. Very different discussion for Europe and the US and others.
Yes.
Well.
Yeah. Okay.
Great. And then one really quick one that, of course, we had come in was, timeline for submission with both FDA and EMA.
Yeah, we're working together with Synthera on the submissions, and the anticipated date of NDA submission for FDA is first quarter next year, so about six, 8 months from now. And we're just getting all the pieces in place to do that.
Great. Well, thank you very much. We really appreciate you coming in and sharing some of that data with us here at conference. We're going to step away for just a second to highlight some PPMD-funded research and be right back.
Hello, my name is Allison Barnard, and I'm a research assistant professor at the University of Florida. I've been involved for several years in the imaging DMD study led by Dr. Krista Vandenborn and Dr. Glenn Walker. In that study, we've developed measures of muscle health in DMD using magnetic resonance imaging, or MRI. These MRI measures have helped us learn more about how the muscles change over time in Duchenne, and now some of the measures are even being used in clinical trials to help determine if investigational drugs are improving muscle health. Using some of the MRI techniques developed in imaging DMD, we will be performing a pilot study in female manifesting carriers of Duchenne to better understand how muscle health is similar to and different from that of typical male DMD. In addition to cardiac changes, female manifesting carriers may have skeletal muscle symptoms such as weakness, fatigue, muscle pain, and poor balance. The degree of symptoms in manifesting carriers can be variable, affecting different people in different ways. There's still a lot to learn about the muscle changes experienced by manifesting carriers of Duchenne. Our limited understanding of muscle pathology has led to gaps in knowledge that create a barrier to inclusion in clinical trials, restrict access to therapies approved for male DMD, prevent development of therapies and interventions specific to the unique needs of manifesting carriers, and limit the ability to develop clinical practice guidelines. With the support of PPMD, we will use magnetic resonance imaging of the skeletal muscles combined with clinical tests and questionnaires to learn more about both children and adult carriers of Duchenne who manifest symptoms. The goal of the study is to develop a natural history data set that can be informative for drug developers and clinicians to improve treatment options for this group of individuals. Thank you to the DMD community for your enthusiasm in participating in research.
Hi, my name is Renzi Han. I'm from the Ohio State University Wexner Medical Center. The PPMD funded project for us is the base editing therapy for Duchenne muscular dystrophy. The purpose of this project is to develop a base editing therapeutic strategy for DMD and determine its efficacy and safety in preclinical animal models. And specifically, we would like to develop a rapid model of DMD for the reason that the mouse model of DMD does not fully recapitulate all aspects of Duchenne muscular dystrophy. So recently, we engineered an improved adenine-based editor, which we refer to as IAB-NGA. And we devised a split strategy for adeno-associated virus packaging and delivery, and demonstrate the high efficiency of cardiac gene correction in a mouse model of DMD. And this work is recently accepted for publication in Nature Communications. And we also developed a rapid model of DMD with engineered nonsense mutation in exon 51 using base editing in the zygotes. And currently, we are working on testing the IAB-NGA base editing therapy in the DMD rabbits. IAB-NGA base editing can be used to correct point mutations, and it can also be used to induce exon skipping for treating DMD. The IAB-NGA base editing does not cut genomic DNA, and it induces permanent genetic code conversion, and thus has long-lasting effect. And this could potentially benefit DMD patients and other genetic cardiovascular diseases as well. I would like to thank all members of our laboratory, and special thanks to our DMD community. Without your generous support, this work cannot be done.
All right, welcome back. Our next session is going to be another In the Pipeline series where we look at some exon skipping therapies that are using some conjugates to help aid in delivery. So first we're going to hear from Dr. Scavina, and then we'll introduce the panel.
Dystrophinopathies, which include Duchenne and Becker muscular dystrophy, are caused by a variant or change in the DMD gene, resulting in little or no production of the dystrophin protein. These changes can include deletions, duplications, or nonsense mutations that disrupt the reading frame, which is the instruction the body follows to make a protein. When the reading frame is disrupted, it prevents the production of that protein, in this case, dystrophin. Let's look at the reading frame as a sentence. DNA provides the instructions for the protein, essentially like the letters in a sentence. Depending on which letters are missing or duplicated, the sentence may be completely changed and not make any sense. Or it may make sense, but not exactly like the original sentence. Therefore, the instruction to make the protein is altered, and either no dystrophin or reduced dystrophin is produced. This is seen with deletions, duplications, or smaller point mutations. If letters are missing, called deletions, exon skipping allows some letters that border the original deletion to be skipped over to correct the sentence and allow some protein to be produced. Antisense oligonucleotide, or ASOs, are single-stranded pieces of DNA that are used to restore the reading frame through exon skipping. If we can restore the reading frame or correct the sentence, we can produce dystrophin again, although it may be modified. The protein may be smaller and the amount may be less than typical. New technologies are being developed to aid in the delivery of exon skipping therapies such as ASOs. These newer technologies act as helpers to the ASO to increase the targeting and delivery of the ASO to muscle itself. This could potentially increase the amount of drug delivered and increase the amount of protein produced.
All right, we're now going to be joined by our panelists for this session. So joining us today, we have Elizabeth Ackerman from Avidity Biosciences, Ash Dugar from Dyne Therapeutics, James MacArthur from PepGen, and Elizabeth Smith from Sarepta. So if you have questions, please feel free to submit them to the comment box below. We're going to have a Q&A after we get through all the presentations. And with that, we're going to hear first from Avidity Biosciences. So Elizabeth, you can take it away.
Great. Thank you so much, Eric and Ryan and PPMD, and for the opportunity to be part of this great panel. And I've been really enjoying the conference. My name is Lisa Ackerman, and I'm the therapeutic area head at Avidity. Next slide, please. This is our forward-looking statements. Next slide. So our mission at Avidity is to improve the lives of people affected by serious diseases by advancing a new class of targeted therapeutics. And I just want to take this moment to say that we are truly honored to be able to serve this community. And we know how important it is that we understand each of your personal perspective and your personal journey with DMD for us to better help serve you. And we are also incredibly grateful to PPMD and other patient advocacy groups that enable companies like us to make these deeper connections with your community. Can I next slide, please? So you just heard from Dr. Scavina that DMD is often caused by deletion or mutation in the dystrophin mRNA that disrupts the reading frame. This does not allow the cell to make any dystrophin. Some of these mutations can be restored, have the reading frame restored by skipping a specific exon in the dystrophin RNA. When this happens, the cell makes a slightly shorter dystrophin protein, but one that is functional. So at Avidity, we are developing 3 different drugs to treat DMD, and they all work by skipping an exon in the dystrophin RNA. The first one is called AOC 044, and it works by skipping exon 44. Now we hope to take this drug into the clinic next year in 2022. The second drug skips exon 45, and the third one, exon 51. So taken together, these 3 drugs, we can address about 30% of the DMD patients who have mutations amenable for exon skipping. Next slide, please. So at Avidity, our drugs are made with basically 2 building blocks. One is an antibody, and one is an oligonucleotide, or a PMO. A PMO is a short piece of DNA, single-stranded, that can work in the cell by causing the cell to skip a specific exon in the dystrophin RNA. Now, there are 4 approved PMOs to treat DMD, approved by the FDA. And these drugs have some limitations in that they can't get into the muscle and the heart cell very well. So at Avidity, we've tried to solve this problem by taking the PMO and linking it to an antibody. Now, the job of the antibody is simply just to take that PMO and get it inside a muscle and a heart cell. Now, antibodies are naturally occurring proteins that are made by your body to fight infections, but they're also commonly used as drugs. And the reason is that they have a property to be able to specifically target a single protein. Can I have the next slide, please? So this slide shows a little bit more how our technology works. We've taken an antibody that binds to a receptor that's found on the outside of a muscle or heart cell called the transferrin receptor. When that antibody binds to the transferrin receptor, the receptor pulls the antibody and the PMO into the cell. Once inside the cell, the PMO is released and it can do its job. So its job is to find the dystrophin RNA, bind to it, cause it to skip an exon, so it restores its reading frame, and therefore more dystrophin protein can be made. Next slide, please. So I'm just going to show you one slide of preclinical data that was created by our researchers. In this experiment, the researchers took the MDM mouse model, which is a mouse that has a mutation in its dystrophin gene that can be fixed, the reading frame can be fixed by skipping exon 23. So what the researchers did is they divided these mice into 3 groups. In the first group, they gave saline or a placebo, and that's shown on the bar on the far left where you can't see it very well. The second group, they gave a PMO. That PMO was able to skip exon 23 of the dystrophin RNA, and that's shown by the blue bar in the middle. And then in the third group of animals, they gave that exact same PMO, but this time it's attached to the transferrin antibody. And that's shown in the green bar on the right. And the taller the bar, the more exon skipping was observed. So you can see that there was about a 50-fold increase in exon skipping when you attached the transferrin receptor to the PMO. So the transferrin antibody. So the antibody did its job. It delivered the PMO into the muscle cell, and the PMO did its job to make exon skipping occur. So I can have the next slide, please. So at Avidity, we're super excited about the potential to use our technology to treat DMD. And we're planning to take our first drug into the clinic, AOC 1004, that will skip exon 44 next year in 2022. Now, our drugs will be given by IV infusions. And for these programs, we think it will be about once monthly. And we will continue to partner with PPMD and the DMD community to give you information as we have it available. But we would love to hear from you, and please reach out with any questions or comments that you have for us simply by emailing patients@aviditybio.com. Thanks so much, and back to you, Ryan.
Thank you, Elizabeth. I'm going to just throw one question at you for clarification on your antibodies targeted to TFR1. You mentioned that. Can you describe what the distribution may look like? Is it just getting to skeletal muscle, or are you hopeful for other types of muscle?
Yeah, so we have shown that by using this antibody to transferrin, that we have really good delivery of the oligos that we're delivering to skeletal muscle, smooth muscle, and heart tissue.
Great. Wonderful. Thank you for that clarification. We're going to move on next to Ash Dugar from Dyne. Take it away, Ash.
Hi, thank you, Ryan. Hi, I'm Ash Dugar. I'm the head of Global Medical Affairs at Dyne Therapeutics. First, I want to thank PPMD for the opportunity to share Dyne's vision with you today. I always feel privileged to have the opportunity to speak directly with the communities and families living with the diseases for which we're developing treatments. Next slide, please. So at Dyne, our mission is to bring life-transforming therapies to patients with serious muscle diseases. And at the forefront of that is our goal to advance treatments for Duchenne muscular dystrophy, which, as we all know, is a condition that still has profound unmet needs despite the current available treatments. Our mission is truly core to who we are and what we do every day. Next slide, please. Well, you've heard a great deal around PMOs and exon skipping in Elizabeth's talk and in the talk before that. So you've heard a lot of great information already that I will try not to repeat. As you all know, patients with Duchenne don't make dystrophin because of a mutation of the DMD gene. And PMOs use a mechanism called exon skipping that you've heard about to skip over the sections of genes with the mutation that cause Duchenne. One of the biggest challenges to having effective therapies is the ability to deliver enough of the PMO into muscle tissue to drive robust exon skipping and adequately increase dystrophin. And it's a challenge that Dyne is committed to overcoming. Next slide, please. So this slide provides an overview of our technology. Our FORCE platform includes a specific delivery mechanism called a Fab, or an antigen binding fragment, which is a small section of a monoclonal antibody. And we've specifically engineered to bind with a high affinity to a receptor called the transferrin receptor 1, or TFR1 receptor. And this receptor is highly expressed across the surface of muscle cells, as you just heard. So the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter cardiac, skeletal, and respiratory muscles. We link our Fab to the PMO with a linker. Once the Fab brings the PMO to the muscle cell, enzymes inside the cell cut the linker, and the PMO is free to drive exon skipping to target the genetic basis of Duchenne. So FORCE uses the Fab to drive binding to the TFR1 receptors on the surface of muscle cells. And we selected Fabs because of advantages such as their small size, which helps with distribution to muscle tissue, and we can also redose with this technology. So this approach is intended to take advantage of specific muscle biology, allowing targeted delivery of the PMO and potentially avoiding off-target effects such as in the liver and kidney. As you've heard, currently approved exon skipping PMOs have been shown to produce very modest amounts of dystrophin. And our approach is designed to much more effectively and efficiently target PMOs to the muscle tissue to enhance therapeutic benefit. Next slide, please. So we've generated exciting preclinical data in Duchenne. This slide summarizes the power of the FORCE platform to effectively deliver the PMO to muscle, resulting in substantial exon skipping and increased dystrophin expression versus naked or non-targeted PMO in a Duchenne mouse model, the MDX model. So this mouse model has a mutation in exon 23 that interferes with dystrophin production. These mice can't run as well, have higher CK in their serum due to muscle damage, and have a shorter lifespan than a wild type or healthy mouse. This mouse model enables comparison of our FORCE platform, which you see in the blue squares, with published data from a naked PMO in the red triangle and a stereopure antisense oligonucleotide in the orange triangle. 2 weeks after a single dose of 10, 20, or 30 milligrams per kilogram with FORCE, we observed substantial exon skipping in the quadriceps, up to 66% after a single dose of FORCE. In addition to the quadriceps, we looked at respiratory and cardiac muscles. We all understand how critical treating the diaphragm and heart is for Duchenne. And as you can see for diaphragm and heart on this slide, there is very modest exon skipping using other modalities. In comparison, the FORCE platform achieves very robust and meaningful exon skipping. This increase in exon skipping resulted in the dose-dependent increase of dystrophin protein expression that you see on the right side of the slide. Our highest dose of FORCE resulted in 8 to 10% restoration of dystrophin production with no demonstrated dose-limiting toxicities. In contrast, this naked PMO restored around 1% of dystrophin with a single dose. So this data gives us confidence that we can leverage the TFR1 mediated cell entry to drive the exon skipping PMO into muscles and generate significant levels of dystrophin protein. Next slide, please. So this functional test in MDX mice shows that the dystrophin is actually working to provide the support muscles need. This is a running wheel test, which measures how active mice are throughout a given time period. The y-axis is distance traveled, and the x-axis is the time of day. And between the two vertical dashed lines is the dark period, when mice, which are nocturnal, run on the wheel. The gray circles are wild-type mice. The black squares are MDX mice given a placebo. You can see the difference in activity. The MDX mice don't move as much. Force in the blue triangles restored function in the DMD mice compared to wild-type mice. And this was not at a single time point, as these data show activity over a 12-hour period. We believe these results indicate a treatment benefit in skeletal and cardiac muscles, since MDX mice treated with force are more active over this 12-hour period compared with placebo-treated and naked PMO-treated MDX mice. So we're really excited to do additional non-clinical studies and enter into clinical trials. Our programs that we're progressing are 51 skip amenable patients first, and then 45, 53, and 44 skip amenable patients. Next slide, please. So at Dyne, your voices are very important to us. We have a deep commitment to understanding the community of people living with muscle diseases and Duchenne in particular. We've initiated many programs involving community webinars, conference presentations, fireside chats, patient meetings, and other outreach to understand the disease better directly from the patient community and gather input to incorporate into our drug development efforts. Next slide, please. This is my last slide. I want to thank you again for giving us your time and attention today. And I want to thank PPMD for giving us the opportunity to present our program to all of you and for this great Congress. When Ravi, Beth, and Diana, who are pictured here, and others speak to us and share their perspectives, it means the world to us. And we greatly appreciate their candor and their insights. Lastly, I want to end by saying again, it's a privilege to have the chance to speak to you. You give our work and a significant part of our lives great meaning. So thank you.
Thank you, Ash. One quick follow-up question. I don't know if you mentioned this. Would this? Are you thinking this is going to be weekly, or will it be twice monthly? What are your thoughts right now on that?
Yeah, thank you very much. So we anticipate this dosing being no more frequent than once a month. Okay. So monthly is currently what we are aiming for, and hopefully we can get to a point where it's even less frequent.
Great. If we can decrease that burden, it will be huge for the families. Absolutely. Wonderful. Okay, we're going to now transition to James from PepGen. James, take it away.
Thank you. Thank you for the invitation to speak to everyone here. We've spent a tremendous amount of time thinking about patients and how we can best treat them. And the approach I'll tell you today is, we think, a very novel approach to be able to improve the efficiency of delivery of therapeutic oligonucleotides safely to patients. If I could have the next slide, please. And next slide. So we've already had a description around the approach of exon skipping to go and essentially skip over problems associated with mutations found in the original coding sequences of the dystrophin gene. And what we're doing here, next slide. What we're doing here is taking these similar oligonucleotides, as has been described already and that all of us are developing, but find a novel way to go and deliver them to cells of patients so they can do this therapeutic benefit of skipping over the deletions and mutations in the gene. Next slide. And that's sort of highlighted in the small diagram in the bottom right. That blue sequence is the peptide that PepGen has spent over a decade developing, that has an extraordinary ability to get into all types of muscles in the body and do so safely. Next slide, please. Now, we've generated a lot of mouse data associated with this approach and can go and demonstrate the skipping of exon 23 and the production of dystrophin protein in mice. Mice, unfortunately, have some limitations, however, in terms of their ability to go and predict how well it's going to perform when we ultimately move forward into patients. So we've done a tremendous amount of testing at PepGen using non-human primates as a model, which are a much better model for being able to identify whether or not you'll see efficacy and good delivery with this approach. In this particular study, we're looking at a single dose that was delivered by IV infusion into monkeys. And then we went and looked at different muscle groups: skeletal muscle, the tibialis anterior, diaphragm, and heart. In each of these tissues, we can see extraordinarily good levels of exon 51 skipping, as indicated by the higher bars, at all of these doses, 20, 40, and 60 mg per kg. We were particularly pleased to see this tremendous efficacy in both the diaphragm and in the heart. As everyone knows, the heart and diaphragm in particular are two tissues that we have to see better levels of exon skipping, which will produce more dystrophin protein to have a really dramatic impact on patients moving forward. Next slide, please. This is looking across a wide range of different muscle tissues. On the left-hand portion, different skeletal muscles, in the middle, cardiac tissue, then smooth muscle, and CNS on the right-hand side. As you can see, looking across all these different muscle groups, at all these therapeutic doses of 20, 40, and 60 mg per kg, and that's following a single dose, we see really terrific uptake of the therapeutic oligonucleotide into these muscles. And that's really important if we want to see a really dramatic increase in terms of dystrophin production. Next slide, please. So what I've shown you so far has been a single dose study, but we've also done multi-dose studies, as this will be viewed as a chronic therapy where we anticipate dosing patients once a month. In this particular study, to sort of accelerate things, we looked at once every 2-week dosing and across a wide range of doses. And as you can see here, even at doses as low as 5 mg per kg, and this is not just PMO dose, this is the dose of our drug, we can already begin to see meaningful levels of exon skipping in biceps and quadriceps and diaphragm. As we go to higher levels, we begin to start seeing it in the heart as well. And we can go and dose at these levels safely and efficiently to go and deliver the therapeutic oligonucleotide. And we think this could have a really dramatic impact. We anticipate that at therapeutic doses, we could be producing double-digit levels of dystrophin protein in patients going forward. And that would have a terrific impact in both improving ambulation and life expectancy in the patients. Next slide, please. Now, exon 51 skipping approach in DMD is our first clinical program. It's the focus of the company. And we're currently in preclinical development, anticipating that we'll initiate studies in healthy volunteers in the very beginning of next year. We can move rapidly through these single dose studies in healthy volunteers and then advance into patients with multiple ascending dose studies, getting quickly, hopefully, to doses that will have a real meaningful impact in patients. Our strategy after that is to move into an open label extension and use this data as confirmation of the efficacy and safety of our approach and of ED51 and its ability to help exon 51 skippable patients. Next slide, please. Now, this isn't the only program that we're working on at PepGen. We are actually working on several other exon skipping approaches to be able to broaden our ability to treat patients with Duchenne muscular dystrophy. We also have a program in myotonic dystrophy, where we think our approach and our ability to go and target different muscle groups efficiently and safely will allow us to provide real benefit to these patients. And in particular, our ability to access the CNS may be of tremendous benefit for those patients as well. Beyond that, we have plans to go and expand across different neuromuscular diseases to try and bring therapeutic benefit to a broader group of patients who are desperately in need of real meaningful therapies that will change lives. Next slide, please. And with that, I'd like to thank you for your time, and I'm happy to take any questions you have.
Sure, thank you so much, James. Just a quick question. We heard from the two companies that are using a form of an antibody to help with delivery, and now you're talking about a peptide to enhance delivery. Can you explain to the community more about that and talk about that peptide?
Terrific. Yes, so this is the work of our two academic founders that the company then took several steps beyond that to go and create a really efficient tool to get into muscle cells. So it doesn't use a receptor, unlike the approaches that were described already, but uses a non-specific mechanism to get into muscle cells, but to do so very efficiently. Now, one of the advantages we think of this approach is this is a very small addition to the oligonucleotide. And so we think it'll get really good exposure deep into tissues, deep into the heart, deep into muscles. And we think that allows more oligonucleotide to get into cells, particularly deeper into the tissue. When we go and look at muscles in different animals, we can go and see that we get very good distribution throughout the tissue. And we think that's critically important for this disease.
Great. Thank you, James. I appreciate that. Now I'm going to turn it over to Liz from Sarepta Therapeutics. Liz?
Thank you, Ryan and Eric, and thank you to the audience for joining us today. My name is Liz Smith, a nurse practitioner and interim head of U.S. Medical Affairs at Sarepta. Today, I'll be sharing updates on our PPMO exon skipping program. Next slide, please. Please take a moment to view our forward-looking statements on our webcast. Some of today's updates will be forward-looking, meaning they haven't yet happened, and there's a risk and uncertainty associated with these statements. Next slide, please. Sarepta's experience in Duchenne runs deep, with over a decade of dedicated research. To meet our mission of reaching 100% of individuals living with Duchenne, we have three diverse approaches to Duchenne drug development: RNA-targeted therapies, which is exon skipping, gene therapy, and gene editing. We have several programs that span our RNA platform using our PMO and PPMO technology, including 3 exon skipping products that have received accelerated approval in the United States. We have an active microdystrophin gene therapy clinical program. We have gene editing partnerships in early stages of development. For this presentation, we're going to focus on our PPMO platform. For a more detailed presentation, please see our PPMO pre-recorded presentation in the conference's on-demand library. Please also see our pre-recorded presentations on our PMO pipeline and gene therapy program in the PPMD on-demand conference library. Next slide, please. In the presentation today, I'm going to focus on our PPMO investigational product, SRP-5051, which is currently in clinical stage. We have previously completed an early stage study called a single ascending dose study. Today, I will focus on Momentum, our multiple ascending dose early stage trial for the evaluation of SRP-5051, our first PPMO product in clinical trials. Momentum is a 2-part multiple ascending dose study for ambulatory and non-ambulatory individuals ages 4 through 21. This slide shows a study schematic for Part A of Momentum. The goal of Part A was to evaluate the safety and tolerability of SRP-5051 at multiple ascending dose levels in order to determine the appropriate dose for further advancement. Patients received one of the escalating doses of SRP-5051 shown on this slide, and dosage increases occurred after clearance by the Safety Review Committee. Unlike PMO, which is weekly dosing, SRP-5051 was administered once every 4 weeks, so patients in the trial received dosing every 4 weeks via IV infusion for at least 12 weeks. Today, we'll be sharing data for 4 patients in the 30 mg/kg cohort of Momentum. We will be sharing data from this cohort because it's a dose that we will bring to the FDA to discuss as a target dose to bring into Part B of the trial. Part B is anticipated to be the pivotal trial pending FDA engagement and endorsement. 3 patients were in their late teens and non-ambulatory, and 1 patient was 7 years old at the time of treatment. Next slide, please. To understand if SRP-5051 is operating as intended, there are 2 questions that we ask. Was exon skipping observed, and is dystrophin protein produced after SRP-5051 administration? SRP-5051 dosed monthly at 30 mg/kg achieved 10.79% mean exon skipping at week 12. Compared to PPMO 20 mg/kg dose, there was a greater than 4-fold dose-dependent increase in exon skipping at only a 50% increase in dose. The data also showed that the increase in exon skipping resulted in an increase in dystrophin. The 30 mg/kg dose of SRP-5051 resulted in a 6.55% mean dystrophin protein expression as measured by Western blot. We hypothesized that dystrophin production will increase over time, so we were encouraged that even at this early time point, The data represent a greater than 100% increase in exon expression versus the 20 mg per kg cohort. Next slide, please. Now, I'll talk a little bit about safety. This slide shows the safety experience across all studied doses of Momentum. In the 30 mg per kg cohort, 2 patients experienced what we classified as a serious treatment-related adverse event, which we'll explain more in detail. Patient 1 experienced 2 serious treatment-related adverse events, and patient 3 experienced 1, resulting in 3 total in the 30 mg per kg cohort. 2 cases were of reduced magnesium or hypomagnesemia, and 1 case was of reduced potassium or hypokalemia. None of the serious adverse events were life-threatening, and both patients remained asymptomatic. Next slide, please. So to summarize, in the 30 mg per kg cohort, we detected mean exon skipping of 10.79% and mean dystrophin expression at 6.55%. The patient receiving the most SRP 50/51 doses, 5 versus 3, at the time of biopsy had the highest expression. Their exon skipping was 11%, and dystrophin production measured by Western blot was 12.1%. We hypothesize that exon skipping and dystrophin will increase with longer-term dosing. Future clinical studies will test this hypothesis. In terms of safety, hypomagnesemia is a newly identified risk in the SRP 50/51 program. Most cases were mild and asymptomatic, with some serious cases occurring prior to the implementation of serum monitoring and magnesium repletion. Our analysis so far indicates that hypomagnesemia or low serum magnesium is manageable and monitorable. Markers of kidney function have generally been normal and have not shown any consistent relationship to the hypomagnesemia. In addition to our analysis, we've consulted with external expertise and believe that serum monitoring of magnesium and oral supplementation with magnesium is a feasible approach to enable early detection and management going forward. Our plan is to meet with the agency to discuss this data and plan for next steps in the trial. Next slide, please. For the sake of time, I won't go into detail, but you can find more information on our pre-recorded presentation. We are committed to advancing our PMO and PPMO therapies and supporting the patients receiving these therapies. We are continuing to use data collection to inform next steps for subsequent exon skipping trials. And we want to thank you, PPMD, for having me here to speak today. And I look forward to being able to respond to some questions. Of course, if you have any questions, please don't hesitate to reach out to our patient affairs team at advocacy@sarepta.com. Next slide, please. Thank you, and back to you, Ryan and Eric.
Thank you so much. Just one quick question before we open the panel. When can we expect to see some of the longer-term expression data to get a better sense of the average level of expression? I know timelines are tough, but we always got to ask. It's our responsibility, so.
Yeah, so more to come on that, hopefully sooner than later. We're working with the agency now to regarding timelines for restarting after the hypomagnesemia occurred.
Thank you.
Great, thanks. So yeah, we're going to move to the open Q&A. So if you have questions, again, please submit them in the box below. And so actually one of the first ones we have in is for Sarepta. So obviously you're working with this PPMO strategy. It's kind of a two-parter. So if it's successful, and you're able to take it forward and get approved, is this something with your already approved products for 51, 45 that would all be transitioned over to this PPMO strategy?
So for right now, our goal is at Sarepta to advance. Right now, at this time point, we don't have a specific timeline for additional exon skipping therapies. We're learning from our 50/51 Momentum and our Mission High Dose study. And as we get more information, that will help inform the direction moving forward.
Okay.
It's an important question that we are actively trying to address.
Yeah, because the follow-up question we had was from a family specifically about one of the ones that's been in the pipeline for a little longer, like skipping exon 52, and they were wondering, is this something where we're going to have to wait and see an approval for the PPMO before that would get targeted, or if that's going to be, like you said, maybe based on what you learned from the Mission study and the high dose about how that goes essentially.
So as I said, we're waiting to inform based on the results of Mission and Momentum as we expand into the other exons.
Great. Thank you.
But our plan is to, continue to develop and expand into other exons.
Thanks. And then, so we saw some data from PepGen looking at some of that distribution and exon skipping in non-human primate models. For Avidity and Dyne, are you thinking about looking at other models besides just the mouse before you enter into your clinical trials? And this could be for all three of you. Is there any interest in looking at some human tissue before you kind of make that next step to first in human? So I'm going to start with you, Ash, because you're at the top of my screen.
Yeah, sure. So in order to, so the answer is yes. We will be looking at non-human primates. It's important data to collect in this particular model to look at the type of data that you saw in terms of tissue distribution and skipping. For those who may not be aware, there's not a dystrophic model, a dystrophic non-human primate model. So you can't look at dystrophin production per se. These are healthy, non-human primates. But it does give you very important tox information and other information that you would want to collect. And so we are planning on those studies, and those studies feed into what we would call an IND, investigational new drug application, that we submit to the FDA so that we can start our clinical program. So the answer is yes. In terms of patient cells, just very briefly, we have done that study, in vitro studies in patient-derived cells. I didn't show it here, but we do have some of that data on our corporate website.
Great, thank you. And Lisa, for Avidity.
Yeah, great. It's basically the same answer. Non-human primates are a really important bridge species, especially with oligonucleotide therapies. They've been shown to be very good for investigating the PK and PD properties of these oligo drugs. So absolutely, we are doing non-human primates, and it will be part of our package for our IND as well. And given, just as Ash said very nicely, given that there's no dystrophin models for the exon skipping that we're looking at that are easy to get to, we have really relied on human myotubes from patients who have donated their myoblasts, and we've done a lot of experiments showing that our different exon skipping drugs work in these MDM-derived myotubes. So yeah, that's also a really important piece of our package.
Great, thank you. James, any additional data you're looking to collect before starting that first? And I believe with yours is going to be an adult population, correct, for the same study?
Yeah, I mean, Ash and Lisa are correct. You can't look at dystrophin production in these models, but in addition to safety, you really get some extraordinary data in terms of biodistribution and also efficacy, because you can look at exon 51 skipping. And obviously more exon 51 skipping results in more dystrophin protein. That's how all of our drugs work. So we think actually it's actually a critical indicator to say, is this really going to move the needle in a really fundamental way for patients? So for us, we think this is the next step that we've now completed. We are finalizing our toxicology package that will be part of a regulatory filing. And with that, we look forward to actually getting data next in humans and moving as quickly as possible and as safely as possible into children with DMD.
Great, thank you.
So I'm going to ask the question that we hear so often from the community. Why do we always start with exon 51? It's a crowded field. There's a lot of activity. It's always targeted. The community needs to know, is it a business decision? At the end of the day, we have patients waiting. And I'm also worried about even recruitment. We're going to have to go outside the US possibly. So I think the community deserves to know an answer to this. So I'm going to start with Elizabeth Ackerman. Put you on the spot first.
Well, actually, good news is we aren't starting with exon 51. We're starting on with exon 44, which of all the exon amenable patients, that's, I think, has been one of the most underserved in the community. So we are super excited that we're going to be able to have our first drug in DMD be for that section of the community.
Great, but 51 will be followed by that before the rarer exons, correct?
We're starting with 44, and that's followed by 45 and 51.
Correct. Okay. So it's still in the beginning of it, and we are grateful that 44 is being targeted. And I I want to stress that that is an underserved space, but the rare ones are still followed down the road. Okay, Ash, I'm going to turn to you.
Sure. So it's a great question. There's still huge unmet need in all of these, with all of these mutations. 51 is the biggest population, and it's still, there's still a lot of need for improvement, frankly. And so, we are targeting 51 first, with 53, 45, 44 behind that program. And you're right. It is a crowded landscape for clinical trials as well, and we are going to be conducting a global development program for our 51 development.
Great, James.
Yeah, first let me just say kudos to Lisa and Avidity for going into 44 first. I think that's terrific. And just to follow on what Ash said, we think that the trailblazing that Sarepta has done is extraordinary, and we're all looking to do better. And we really think at PepGen that our approach really can do something extraordinary for patients. If we could produce double-digit levels of dystrophin protein after 3 months of therapy, that would be a game changer. So why start off in 51? For us, actually, it's that there's so much validation, if you will, in terms of the clinical development path, the outcome measures. And for us and for investors, we find that it's easy to go and say we can benchmark ourselves against those who have gone before us and say, how much better are we doing? And that allows us to then really drive forward into other approaches. One of the great things, though, about this peptide-based approach is very rapidly, we can go and do a plug-and-play approach, take the same peptide and put it on a different oligo and move from one indication to the next indication to the next indication. And that's what we're building the team to go and do. We think this is what the community deserves.
Yeah, and just as maybe a follow-on to that, so since there is some overlap in terms of the exons that we're targeting, and there's going to be, there's only so many patients with those mutations, and we do see a lot of trials kind of target that same early ambulatory 4 to 7 range. You're probably early in your discussions around clinical trial design, but are you thinking about other populations that maybe don't have or aren't given the opportunity to participate in these trials as early? As a way to kind of power your studies. So James, I'm maybe going to throw that back to you real quick.
Yeah, I think that's a great point. And I think we do need to go and broaden how we're looking at patient populations and accessible patient populations and patient populations where we can learn a tremendous amount clinically. And we're fortunate that Jane Larkindale has joined us at PepGen and is a great thought leader in terms of thinking about how do we do those sort of trials and get to those sort of patients and provide a broader benefit. Great question, though.
Great, thank you. No, and we miss Jane already from her work in de-risk, so good.
Oh, she hasn't left you.
No. We know where she lives, so.
And we actually have some other questions coming in, so I think I'm just gonna kind of bounce around real quick. So, Liz, with Sarepta seeing kind of the low magnesium levels in response with treatment, is that something, you talked about kind of adding that to the protocol, is that something patients should be maybe expecting? Is that something we're just going to see throughout, the long-term treatment with this drug that becomes kind of part of that process of the infusion that you're gonna be have some additional, treatment layered on there?
So for part of the clinical trials, we will be adding additional monitoring for magnesium to monitor for hypomagnesemia. We believe that this is monitorable and manageable. We've sought out expert review from nephrologists, but we will be continuing to monitor in the ongoing clinical trials, and we'll know more as we progress in Part B for momentum.
Thank you. And then it's early, obviously, so hard to tell some of this information with your products in Duchenne, but questions about with these other ASOs using conjugates, potential safety or side effects? Are there things we can learn from other indications or other places where there's monoclonal antibodies being used or the Fabs being used that we can maybe take as you kind of are planning your trials as well? And Lisa, maybe I'll start with you.
Okay, yeah, great question. And I think, monoclonal antibodies have been used now for a long time. There's a lot of drugs out there. We know how to make them. We know how to make them safer. So it's in the same thing with oligonucleotides. So we're taking two very well-known, well-used modalities that are already used in many drugs that are out on the market and hooking them together. So of course, when you hook them together, that makes it new and you never know what you're going to find. But at least the two pieces we have a lot of confidence in. And we also, by using the same antibody for all of our neuromuscular programs, what we learn from one program will really be helpful for the other program. So we will be going into the clinic for DM1 using this monoclonal antibody that we're using for DMD. So that was going to be really important to form us on the safety. But so far, all of our non-clinical data looks very good.
Great, thank you. Ash, if you'd like to add anything.
Yeah, just a little bit. So, as Elizabeth said, there is a good deal of information out there on antibody drug conjugates. So I'm sure all companies are looking at what information is in the literature and medicines, different antibody drug conjugates that are on the market now. At Dyne, we're taking a slightly different approach, or a very different approach in terms of taking a portion of that antibody, and we think that'll confer certain advantages. So we're excited to see how our conjugate performs as we move further along. And as Elizabeth alluded to, we have a very modular approach, and so we can interchange PMO that skips 51 or PMO that skips other mutations, which allows us to move our programs through non-clinical development very quickly. And then we are also focused on other diseases like myotonic dystrophy type 1 and FSHD, where our Fab and linker remain constant. And it's that therapeutic agent that we can interchange. And so we're able to move our programs through very quickly because of the modularity of the FORCE platform.
Great, thank you. And I just want to say thank you to all the panelists. We appreciate Sarepta sharing some of your data, and with the other companies, talking a little bit about what you've been working on to date, and kind of looking forward to your clinical trials as you all advance your program. So look forward to speaking with you all in the future. With that, we are going to take a moment to spotlight a few more PPMD programs, and we will be back with our panel on facilitating independence.
Hello, I'm Michelle Grimm from Michigan State University, and I'm the PI of PPMD's grant to study the effectiveness and performance of a motorized exoskeleton system for individuals with DMD. I'm working with Blake Matthey of Talem Technologies and Richard Fowles of Really Useful Robots. As we are all aware, at around age 15, young men with DMD experience decreasing arm strength that makes it impossible to lift their arms against gravity, and greatly reduces their ability for independent manipulation. We are working with a pair of motorized exoskeletons developed by Talem and Really Useful Robots that senses the user's intention to move and amplifies their remaining arm strength by up to 40 times. We are very pleased to find out that despite not having full use of their arms for many years, all of the participants use the motor control they learned prior to age 15 and regain nearly full arm range of motion immediately. Through a cell phone app, the users can periodically adjust the amplification to accommodate future loss of strength, and we anticipate that a useful life of the exoskeletons of 10 years. Our PPMD grant allows us to equip 30 young men with a pair of exoskeletons so they can have use of both of their arms. The exoskeletons are mounted on the user's wheelchairs and draw power from the wheelchair batteries. Our participants are teaching us what they can do now with their newfound manipulation. This includes doing everyday things like adjusting glasses, brushing teeth, or simply scratching an itch. These exoskeletons open the door for engaging in hobbies like model car racing, playing the piano, or building with Lego. With increased arm movement, the guys can expand their intellectual horizons with the development of microcontroller projects in engineering classes, and the use of computer-aided design and 3D printing. The increased range of motion is accompanied by the ability to effortlessly lift items weighing up to 3 pounds. PPMD has made it possible for us to conduct a long-term, multi-user study of how restored arm function can improve daily life in the community. We have shipped 17 systems so far and are in the process of building the final 13 for delivery, 2 per week, through July. We are grateful to the Parent Project Muscular Dystrophy for supporting the first major translation of exoskeletons to assist young men with DMD. We are also exceedingly grateful for the opportunity to work closely with 30 wonderful young men and their families as we learn how to improve the technology so that it can play a useful and important role in their lives.
Hi, everyone. I'm Rachel Poisky, mom of Joel, and co-coordinator for the South Texas Connect Group. September is coming soon, and it's Duchenne Action Month. Every day in September, PPMD will provide simple things you can do to raise awareness and funds to join our fight to end Duchenne. You'll also get updates on research and care and ways you can advocate both locally and nationally. Plus, September 7th, the global community will celebrate World Duchenne Awareness Day, and you don't want to miss that. My family participates in Duchenne Action Month through Coach Secure MD, a partnership with the American Football Coaches Association. We also host a local event of Coach Secure MD at a local brewery or restaurant every year to raise funds and awareness, and it's a great opportunity for our friends to come out and support us and our family. From creating a simple fundraising page through Facebook or hosting your own event at a local venue, you can be part of Coach Secure MD. There are so many ways that you can get involved to raise awareness and to raise critical funds that we need for research. Visit the PPMD website today for more information on how you can get involved in Duchenne Action Month, specifically in Coach Secure MD, but in lots of other programs that are out there. Let's get together, let's raise awareness, and let's raise money for research. Duchenne Action Month.
Welcome back. It was really great spotlights and good to see the progress on the exoskeleton.
Yeah,
really
it's
exciting.
fantastic.
And actually transitions well into our next session. Our next session is on facilitating independence. This discussion will feature members of our community along with clinicians. The session is going to be led by Kathy Kinnett, PPMD's Advisor of Clinical Care, and Pat Motion, Coordinator of Adult Advocacy and Engagement and co-leader of our PAC. I'm going to hand it over to them now officially.
Thanks so much, Ryan, and thanks everybody for joining us. We're really excited to have this discussion today, and everyone who knows me knows that this is a really important topic and for me and for everyone in this community. So I'd like to start by just introducing the members of our panel. So Bailey, do you want to start?
Hi, I'm Bailey. I'm part of the social work team at Newborn's DuPont Hospital for Children in Wilmington, Delaware. I'm the social worker that works directly with Muscle Clinic, and we see children from birth to 21.
Thanks so much, and thanks for being here. Christina?
Hi, I'm Christina Trout. I'm the nurse for the Neuromuscular Clinic at the University of Iowa. And I was also a part of the CDC's transition panel, so was involved in creating the transition toolkit. I see folks from the time of diagnosis through till the time they're old. So I'm very happy to be involved in talking about transition because it's been something near and dear to my heart.
Thanks so much. You've done a lot of work in this area, so we're glad you're here. Austin, do you want to introduce yourself?
Yep, I'm Austin LeClaire. I'm a member of the PPMD PAC, and I also am a consultant in the healthcare field.
Thanks so much. And Rachel?
Hi, I'm Rachel Poisky. I live in Houston, Texas, and I am a Duchenne mom. My son is 18. He just graduated high school and is headed to community college. So that's me.
Excellent. And Pat Motion. No one's seen you yet this conference.
Well, I've been, I've been behind the scenes with some outstanding closed sessions with a bunch of Duchenne and Becker guys. I am 48 years old. I live in New Hampshire. I have actually limb-girdle muscular dystrophy, and I am married with 2 kids. I am also a full-time music teacher, and along with Austin and my colleague Amanda Wilkerson, I help run the PPMD PAC, which is the Patient Adult Advocacy Committee. It's a group of 24 guys with Duchenne and Becker, and we kind of help elevate everyone's life through a bunch of holistic different things. We are trying to make our voices heard, especially today with facilitating independence. So glad to be on this call with you guys, and good to see everybody out there in TV land.
Thanks, Pat. So to kick this off, the title of our talk is Facilitating Independence. Would you maybe start talking about what independence is and why we think this is important?
Sure. Well, facilitating independence is something that all of us need to learn how to do, whether or not you have muscular dystrophy or not. So I think one of the main things to remember as we launch our panel is some of these independent steps are going to be helpful for everybody, but especially as you transform from being a child into a grown-up. Somebody very, very succinctly put it to me last year that has Duchenne. He said, this is the generation we were hoping for. All of us living longer, and now we're here. So we have to deal, and we have to figure out a way to help these men live independent, fulfilling lives. So I think it's important to remember that some of the takeaways here are going to be good for everybody, whether you have an affected child or not. We have to put everything in the same basket, and we're trying to raise independent human beings. So that's going to be our goal. And I'll send it back to Kathy. You can kind of tell everybody how this is going to work for over the next 30 minutes or so.
Yeah, so we're just going to have a discussion, and I think we're going to start off with Rachel. So Rachel, we always want adults to be independent, and we want them to be productive members of society and fulfill their dreams and their wishes. But I don't think independence necessarily starts at 15. So can you talk a little bit about how parents can help young children start to facilitate independence, especially in light of some physical limitations?
Yeah, sure. I think it's something now, having an 18-year-old, I want to go back and tell all the young parents, listen to me. I want to tell you what your future is like. And, when your child is diagnosed, you're so consumed with the healthcare and the clinical trials and the stretching and your grief, that helping them to be independent is the last thing on your mind. And I get it. I've been there. But I think it's so important, especially now with all our clinical trials and everything that's happening, and our boys, and girls living longer and having productive lives. We've got to start to think about independence. And that starts as early as possible, just like with any, child. So that's something, having an 18-year-old, I think, wow, there are so many things I could have done along the way. I was consumed with everything else, as I should have been. We can never, kick ourselves with Duchenne, right? But I just want to say to the younger parents, please start thinking about some things that you can do with your child, because it is important, not just for them to be independent, however they are, but for their self-esteem, for their well-being, for their happiness. So it's also part of their mental health to work on that independence piece.
Thanks. Yeah. So Bailey, there are lots of behavior issues in some kids with Duchenne, some boys with Duchenne. So it's hard enough to parent a child without a lot of behavior issues and try to get them to do things that you want them to do that maybe they don't necessarily want to do. So how can you help parents, or how can we help parents work with behavior issues to also help facilitate independence?
I think just getting that support in place for you and for your child, whether that's helping them build friendships at school, and really seeking professional help when necessary, and including the peers as well as the professionals in their lives. They can have open conversations with their teachers, and that can really introduce that sense of independence, or involving them in their medical appointments too. It's not going to be easy, and each situation is unique to each person. So you know your child best, and just pushing them a little bit further each time will foster that independence.
Parenting, that's not always easy to do, is it?
No, and definitely not with behavioral issues either. But your child is your child, and you're going to know what they need and how to support them throughout their life.
So Austin, you're a pretty independent guy. What are some things that your mom did that helped you become independent and maintain your independence?
Yeah, so when I was really young, around the age of 5, when I was diagnosed at the age of 3, but since then, that whole time frame, my mom had worked at restaurants, so she was a waitress, and so we had to spend a lot of time. She worked at a marina, and we had to spend a lot of time entertaining ourselves. So I think that definitely helped with the independence, because she encouraged us to do stuff on our own. And in our household, we have always been required to make our own food. So everyone in my household can cook. My brother Max, luckily, because he got an X on skipping soon enough, he's still able to make his own breakfast. It takes him a lot longer, but he still can do that. And so really in my household, it's expected that we can do the things that we can do on our own. We all have chores. My brother Max has chores. He has to fold laundry and stuff, because we find the chore that we're all capable of. So a lot of the time it was just us entertaining ourselves. We had a lot of nannies. So a lot of the time we had to be able to entertain ourselves and figure that stuff out. But with most of the independence, it was really just letting me do it on my own, because I enjoy being independent. But I've learned also not everyone is cut out to be as independent as I am. And it's completely fine to not be super independent. But what we do need to do is give people all the resources they need to be independent if they seek that life.
Yeah, I think, Austin, that's great. And I will add, I was diagnosed at age 12, and along with the diagnosis came the same fears and the same worries about the future. But I think it's very important to note that my parents did not build a cocoon around me. They were very open and honest with me. And they also said, at some point, it's going to be your life and you're going to take over your own life. So we need to get that started immediately. I mean, they didn't say it in so many words, but the cocoon thing is huge. As a teacher of middle school students for many, many years, there's a concept of learned helplessness. And I think although it sometimes can be an elephant in the room and parents don't want to hear about it, especially if you have a disabled child, it's easier to ignore that, thinking, oh, it'll be easier if I just do this for him or her, or I'm going to help there, or I'm going to speak for them. What you're really doing is creating a secondary problem. And the problem is that the child is going to grow up thinking that they don't really have a voice and they can't be independent. So then all of a sudden when they're 18 and it's time for them to make their own appointments and this and that, they're going to not have any of the skills because it hasn't been seeded and fostered at a young age. So what Austin said is very important. I also had chores and household tasks. It was not focused on, hey, you have muscular dystrophy, look at all the things you can't do. It was, hey, you have muscular dystrophy, here's the facts, here's what's going on, age appropriate, of course. And then it was, okay, here's the things that you can do. So go do them. And that definitely, I think Austin would agree, led us to becoming, more independent adults. And as he pointed out gracefully as well, everyone's definition of independence is different. I want to just add one more thing about the household chores since I'm rambling, but talking to other Duchenne guys, some of it is, maybe they can't cook, but they can help with the grocery list. There's nothing saying that you can't go through the house with your kids and figure out, hey, what do we need? Make the shopping list, help me figure this out. Or, once a week, you figure out dinner. Well, okay, so we're calling out. You got to research a bunch of restaurants, figure out what everybody likes, plan the meal, figure out the money. I mean, it's all that kind of stuff that you probably just say, oh, it's just easier for me to do it. Hit the brakes, involve everyone in the household. Rachel, I see you nodding vigorously. You want to throw in here?
Yeah, I think, I imagine some moms are and dads are saying, oh gosh, now I have to add one more thing to my list. But I don't think it's on us to be overwhelmed about creating independence. For me, I try to think about what is one thing I could do each day to foster independence, so it's not the whole picture. And, like Austin said, there's some naturally, just in general, there are some, kids that are going to be more independent than others, but you have to keep pushing, especially I think with the Duchenne community, there's a tendency to kind of turn inward in a lot of different ways, socially, physically, all those things. And it's a little bit of continuing to push out and pushing your child outside of their comfort zone, just pushing them a little bit more toward independence every day. And like you said, finding things that they can do. There are things that every single person with Duchenne can do to be more independent than they are today. So just to take it one day at a time and try to keep moving. It's not solving the whole picture, but it's moving every day a little bit more toward independence.
So one of the things that goes along with fostering independence in my mind is fostering socialization as well. So does that go along? Does that make sense to you all as well, Christina?
Yeah, I was going to comment that I think all of these concepts of like when to start establishing independence begins at school, even preschool. That there's a lot of practice that young people learn at school about becoming independent, and it's also where they learn to socialize with others. When I talk with parents, I ask what ideas they have about making sure their child is connected with their peers. I know that there are sometimes barriers to visiting other person's houses if they're not accessible, but there are ways that you can help think about how to help them, either have them over to your house or ways that they could meet in the community to go to a movie, go to the mall together. So be thinking about ways to keep your young person connected to their peers and be practicing that independence even from the time of early school years. I would also mention that I'm from the healthcare background, so in terms of activities of daily living and health, I also think that at school we learn those very first lessons about how to direct your care, how do you prefer to be cared for when you're working with a one-on-one attendant at school. So once your school needs are identified that you need some support, it's a great time to learn how to tell others what it is that you need and be autonomous in explaining that to others.
I have no comment. I'm very antisocial.
And not everyone has a problem remaining connected with their peers, but I see a tendency for some of the guys to lose connections at certain times of their life. And really during the teen years, I can see sometimes that happening where everyone's going to the football game or at school, or everyone is doing a certain activity or playing a sport. Many of the guys in my group have been managers for their teams at school. Many of them have been on the newspaper or yearbook, taking photography, contributing to the editorial support on their school activities. Those are things that not only keep them connected, but also point them in a direction towards things that they want to do when they're maybe going to college, or what they're going to do after high school. So like all other youth, we have to be thinking, what happens after high school? It's not an end point, it's a launching point. It's time to move into young adult life. And there are many facets to adult life that all of us have to continue working through, not unique just to Duchenne, but I think it's a time to network, find things that everyone likes to do, and how those could be used in the future.
So Austin, a lot of people think that being independent means not asking for help. How do you bridge that gap?
So I think that you always need to be able to ask for help. because I'll always need help for certain things. Like I know I'll never be able to use the bathroom on my own. So I'm always willing to ask people at school to help me use the bathroom. I've never had a student tell me that another student tell me they wouldn't help me. So I think that it's really independence. You have to know when to ask for help, because I've been not willing to ask for help for a while, and it's led to a lot of mistakes. And I think that you need to know, the sooner you can learn to ask for help, the better. I even to this day still feel bad sometimes when I ask for help for things. And that's completely normal. But you have to remind yourself that if I could do this, I would do it. So I think it's really important to ask for help.
What about, of course, it also, it's how you ask. It depends on, it doesn't matter if you're caregiver, family member, or whatever, you don't want to obviously, nobody likes to be directed around and told what to do. But if you explain the situation, I have found, as Austin has too, that the overwhelming majority of people are happy to help you. Everybody likes to help others. And it's how you present it, and it's how you kind of, how you frame the ask. Austin, I do want to ask you though, what about boundaries? You talked about like going to the bathroom and stuff like that. I mean, this might be also a little bit difficult, especially if some of the people on this call have younger kids, but, thinking ahead, what, things like that. How do you deal with boundaries in your life that you need to have as an adult male?
Generally, so when I typically need the help using the bathroom, I just need someone to take my leg supports out and hand me my urinal. So in those typical situations, if I've known someone for even only a day in my class and I really have to use the bathroom, I'm okay with asking them for help. I think that that's not crossing a boundary. But if I need someone to lift me like on the toilet, for example, that's beyond boundaries. I think that that's where I draw the line, because they're going to have to give more help than just handing me some stuff. So I think that if it involves more than just handing me something, then that's too much. Like I'll even ask for someone to take something out of my bag. But if I need you to physically like move me and stuff, that's crossing the line.
Well, and I also think that it comes back end around to the fact that you also began to have these thoughts in your head when you were young, and it was independence in ways that, and when you're young, you can't see the dots connecting yet. But as you got older, then you were comfortable asking people to help you with certain things, comfortable where the boundaries were. And also everybody with Duchenne Becker will tell you one of the things of our existence is we have to plan many, many more steps ahead than an average able-bodied person. So if someone else thinks they can do it in 5 steps, it might be 15 for us. That doesn't stop us from going out and socializing as adults, but you have to think, okay, where am I going? What's the bathroom look like? Is it a public space? Who's going to be there to help me? Things like that. So the boundaries are established, I think, long before you need to come up against them. But again, little things like Rachel was talking about and Christina was mentioning, little things of facilitating independence when you are young, it all sets the stage and sets the table for things that you can now handle when you are older and you're not, again, in the cocoon.
I was going to say too, I think another part of facilitating independence is helping your child to understand and use resources out there. For example, my son's school did a great job of prepping him for his college and contacting disability services. So he did that completely on his own because the school had prepped him and said, that's one of the first things you're going to need to do before you do anything else at your college. And so he's going to a local community college. His independence is he's not able to live on his own right now, but he's doing a community college and he's taken responsibility for that and reached out to some of those services he needs. So I think what we can do as parents, we're always gleaning resources for us, but we rarely communicate those well to our kids or include them in that. Hey, we're using this resource, we're using that resource, so I think that's important too, is to help educate them on what resources are out there to help them be independent.
I agree. Bailey, do you have tools and resources that you share with your patients as they get a little bit older and they start to think about life after high school?
So one of the big things is a lot of my patients have to decide if they want to go to school, like Rachel said, or not. Contacting that school's disability service is going to be key. Also, regarding transportation things, there's different community transits depending on where you live. I know each county is different, and even each state. So like in Delaware, we have JART, or in Chester County. In Pennsylvania, we have Rover that can often work with the families to bring people places. And then it's also connecting to maybe Medicare, Medicaid resources. If you have a case manager through your insurance, that they're really helpful because they can make aware of things that we might not need to pay for either.
Yeah, boy. Go ahead. Yes, please do. You were next.
Well, I was going to tag on to what Bailey was saying. When folks come to visit us and they have back-to-back medical appointments and tests all day long, oftentimes they don't get as much time as I think would be helpful to meet with a medical social worker or the care coordinators at their clinics. I think in terms of the interim, the things that happen between appointments, sometimes the information that you might get from working with your social worker and care coordinators could be incredibly valuable. I would also put a plug in that you can reach out to all of us, care coordinators and social workers, between visits to have some additional conversations or set up appointments to really go through these topics. The transition toolkit, and I wanted to put in the plug for the Education Matters, has a timeline for educational planning. When Rachel was talking about getting ready for college and meeting with disability services, this does not happen overnight. It's helpful to be thinking about learning about what resources are available in your state, in your community, and in your community college or university settings, so that you can know when is the best time to enlist those services, because there may be waiting lists for some of these things. So ask your medical social worker, care coordinators about resources that are available. Make sure that you're having time to get plugged into those. And look at the PPMD Education Matters booklets. Those that are for the teens and adolescents give a little bit of a timeline and a reminder about these things that you'll want to tap into.
Thanks, Christina. So Austin, how do you make sure that you maintain some independence in your social life? How your mom and you work that out?
So actually I have a lot. So I have friends that typically bring me on trips and stuff. So my mom doesn't really have to be involved in it. But this last weekend, I actually went on a trip with my friend, and I had one of my camp counselors from camp. So we've been friends for years. So he brings us on trips and stuff. Last year in March, he brought me to the PPMD conference, and so he's really good at bringing me on trips. So generally, my mom isn't involved when I have social events. It's kind of, I generally have to figure out transportation too. Like lots of the times when it's something near my house, I'll take the train on my own. But it's really, my mom really doesn't get involved when we've had other friends' parents in the past complain to my mom about a plan or whatever that me and a friend were coming up with, and my mom would always say, they're adults, they can figure it out. We don't need to be involved in this. And so my mom really stays out of my social life. She lets me, I have to obviously communicate with her when I have people coming over. But other than that, social gatherings, I deal with all that on my own.
And again, not to belabor the point, but your mom helped you set the table to be able to do all that stuff when you were young. Speaking of young, Rachel Poisky, I know you just turned 30, so you're a very young parent. I want to ask you, I don't know, I'm just, listen, a lot of people out there know me, take me with a grain of salt. Rachel, what about, since you're the mom on this call, and here's another elephant in the room, can you give us an example of, or maybe more than an example of, a tough love moment that you've had as a parent? I know all the parents out there also are there's probably a thought process of the fact that this is all great, but again, that light of Duchenne is still going on, and the kid is very fragile, and I'm not sure what to do, and sometimes you got to throw down the tough love. So give us a couple of examples of that.
I think one example was, and I know this sounds silly, but it was really hard for me. My son didn't want to ask for anything, restaurants, didn't want to do anything. And we were at a fast food place one time, and he said, "I want ketchup, go ask for ketchup." And I said, "Nope, you can go ask for ketchup. That's not my job." And he was like, "Then I won't have ketchup." I was like, "Then you won't have ketchup." And I know that sounds silly, but at the moment it was really, we talk about all the behavior stuff. There was a major eruption in the middle of the restaurant, and it was difficult. It was difficult because I just wanted to go get the ketchup, and I didn't go get it. I think that was one thing. Another thing is, we haven't even talked about siblings, but there was one point where my son was interviewing for a school, and his sister had something else, and I promised I would go to her with that. And I had to send him to this interview at the school with his granddad. And he was like, "I can't believe you're sending me." And I was nervous, but I said, "You know what? I've already committed to your sister, and I have to stay committed to your sister, to what I promised I would do with her. And you're going to have to go do this by yourself." And those are two moments where, I just white knuckled and I said, I have to let go. And I think that's Duchenne every single day. And it's hard sometimes because we have some, boys. I think the other thing we haven't talked about is how anxiety plays into this. A lot of the Duchenne boys and girls with Duchenne suffer with anxiety. And so it's anxiety provoking for them to head toward independence. And so sometimes we want to just, take care of their anxiety and manage that for them. And so we think, well, never mind, they're too stressed out. So I just want to say to Duchenne parents, you're fighting a lot of battles toward independence. It's not just one, and that's okay. Like, it's hard. This is a hard road. Duchenne is a hard road, but promoting independence, sometimes you have, kids like Austin that want to be independent, and sometimes you have some that don't. And that's why I say you just take one step a day toward independence, but go ahead and do those white knuckle moments where you don't want to do it. Every time I've let my son be more independent, he's come out with more self-esteem, more self-confidence, and moved forward. And just his mental health is better. And so I just want to say, for your child's betterment, self-esteem, growth, giving them more independence is critical.
All of that is huge. And I would add, certainly not silly at all with the ketchup. You meet the child where they are. To you, that's a very small thing. To him, it caused an eruption. And other people might say, "Oh, that's I wouldn't even think about it." You meet the kid where they're at. You'll see it'll come out when they're like, "Oh, this is a chance that I can foster independence." And it will help. It will help all the time. And it doesn't have to be anything huge. It's small steps, like you said.
So, Christina.
I was going to mention that not everyone goes through development and steps towards independence at the same rate, too. So make sure to compliment your son and your family when you make small steps. Not everyone is going to be a leap, and not everybody has to get there at the same rate. So don't be too hard on yourself in those small steps.
So, Austin, how hard is it how do you develop trust in people to help you? I mean, you can ask people, but if they haven't helped you before, it's probably a little bit more difficult. So how do you trust that the people that you're asking to help will help you, and how do you help them know what to do? Austin?
You okay, Austin?
Yes. So when you ask questions for help, I think really you just have to be really friendly about it. Like when I have just been hanging out with a friend in one of my college classes that I just met that day, I always say, "Hey, do you mind helping me use the bathroom?" I don't think that asking for help is too complicated. You just have to go in with your intent of getting help. I think it's pretty simple. It's very overwhelming, I understand, to like approach someone or ask someone for help, but you kind of just have to rip it off like a Band-Aid and just do it. Because the worst thing that they're going to say is no.
Yeah, I don't disagree. And speaking of help, when I was in the clinic, I would have lots of parents that would, talk about how tired they were and how hard this is, which it is, and they were. And then I would ask them if they had people coming in to help them, and they would say, "Oh, no. I don't want anybody else in my house," which with COVID, that's a whole different thing. But before COVID, so I don't want people in my house. And then they would say, "But my son, he doesn't want anybody to do this except me. And so it's easier for me to just do it than to bring somebody else into the house." So I'd love to ask that question of everybody. Bailey, how do you help parents manage this so that they can get the help that they need and they don't get burnt out, and guys get used to having help from other people besides parents?
Yeah, so we always talk about home health aides or anyone like that. And it's important to know that you don't need to stick with the same home health aide if it's not working for you, as well as there's always going to be a meet and greet. And if that person isn't working out, or if you just get bad vibes off from the start, you can say no, and we continue to look for different agencies or different people. But then it's also connecting to respite and allotting that through the state. And it could be as simple as getting your parent to come to your house and foster that relationship with your child, and just so that your parents can have a night to themselves. And I'm always checking in to see how the parents are doing, because you can't take care of your children if you're not taking care of yourself.
So one of the things that parents don't often look into is respite because they don't know where to look. Do you have any suggestions for that?
Yeah, so getting connected to your county through the developmental disability services is the number one way to access respite. Your social worker at your clinic should be able to help you fill out the forms, as well as connect you to the right people. It's not a lot of money, but it is something. And a lot of my parents actually choose to then send their kids to camp sometimes. So that's like a nice long break too. And you can use the money how you allot.
So Rachel, how have you all handled people coming into the house and maybe Joel's discomfort with outside agency?
Yeah. Well, we've been lucky that actually the grandparents have served as respite. We were on a 7-year wait list in Texas and finally got respite care. We're still on a 15-year for actual, well, we've been on a 15-year list to get caregivers. So hopefully in a couple of years we'll come off that. But for us, it's been family members. So I think that has been something that's made it easier. So we haven't done any outside out of family members yet, but that's worked out well for us. But I will say too, a few times I've just had Joel be with another special needs family, not even necessarily Duchenne. I know you sometimes can't have Duchenne, but we've provided respite for each other. And because I think you feel like with this another special needs family, it's like, okay, I can trust them. They all know that they need to call the doctor if something's wrong or that kind of thing. So I would just encourage you to look around and see where you can and that also deepens your social connections with other members and people you can support. So you can get creative. I mean, I do think at some point you have to really ask the question, how can I get comfortable with strangers and not strangers, but people in my home? I think the Duchenne community as a whole is not comfortable. Everybody's not comfortable with that. But I think for our kids to become more independent, for us to be able to have self-care, we've got to continue to move in that direction. But I also would say, if you're not quite to that point yet, get creative, because you do need time away. You do need some self-care. So think of some ways that you can do it within your comfort level that will still, be able to foster some independence and give you some help that you need.
I would also just add to everybody listening that this whole presentation is very toe in the water. And with the advent of the Zoom times here and all the stuff online, I've talked to a lot of families that everybody gets, real jacked up at conference, and then it's a long time. It's 365 days to go around the year to the next one. Please, if you're getting a point off of this, we are all more than willing to talk to you about any of this at any time. I feel like a little bit you might be drinking from the fire hose because we're all sitting here throwing rapid fire big topics out at you. But this is an ongoing thing, and facilitating independence takes years. So we on the screen right now, we're all resources for you that you can reach out to in the future. I know Austin has had a lot of experience with hiring personal care attendants, both good and bad. Bailey has a lot of resources, and Rachel's a mom, and I've gone through the journey myself. So please, you can reach out to us as well in the future. I wanted to say that. I also want to ask Austin, another elephant in the room that hasn't really been brought up is, especially a Duchenne boy growing up, you reach the age of 13 and maybe puberty is delayed a little with the steroids, maybe it isn't. How do you deal with privacy? That's another big thing that families get very hesitant to talk about. Young males need privacy from caretakers and parents. Austin, are you comfortable discussing a little bit of those kind of boundaries or things like that? Or I will just throw out, that might be one of the times if you are at the clinic visits with your son and your son's 14, 15, 16, that might be a good time for you to advocate for your child to have you step out of the room. And then your child is talking to the health professionals without you there. Because guess what? They're still a normal functioning male. And mom and dad, there's a lot of things that they want to talk about that they don't want you to know.
Or even privacy at home, Austin. How do you get privacy in your room when you know nobody's going to come in?
Well, so in my house, I'm very open with my mom about stuff. So I've never needed her to leave the room when I talk to a doctor. So I think that's a really good thing that my mom and I have that relationship. But for the most part, I don't need to ask for any privacy. I generally am in my room with the door closed anyways, because my mom doesn't like my hell dog running around.
So if you were going to invite a young woman over, your mom is sitting there, that's fine. Come on. We're talking about the other kind of privacy. No, I mean, you just, advocate for your son to ask them, hey, would you like 15 minutes? It's a normal thing, and nobody wants to talk about, the sex portion, whether it's with others or when you're exploring yourself as you're growing up. But you got to give some of that normalcy and privacy too. Think about even those kind of feelings. You as an adult, you need 10 minutes by yourself, whether you go for a walk outside. I can remember talking to a lot of families when they have babies. It's like sometimes when I go into the bathroom, that's the only 5 minutes I get a day because the kids are up in my face. So your son needs that as well. So even if it's awkward or difficult, please address the privacy issues because it's a needed thing.
I have to chime in even from a healthcare standpoint. There are I get questions from young men because unfortunately in the clinic setting, sometimes they were not offered private time alone to ask those very questions. For example, in the act of self-pleasure, the heart rate goes up and I know I have a cardiomyopathy. Is this a medical concern? That's a real question, and they may not feel comfortable asking that with a parent present. So I would hope that your healthcare professionals are addressing some of these issues. But as a parent, please offer to step out. Even if you don't think your young person has those questions, it's a good time for the young person to start practicing interactions without their mom and dad present. And maybe the healthcare professional would bring up a topic that would not have come up if the parents weren't sitting with them. So it's great if there's an opportunity to have one-on-one time with healthcare professionals without the parents present in that setting. And then there's also opportunities that young men and teens can reach out to their healthcare providers and ask those questions, as like you say, in Zoom. We have email, we have all kinds of private electronic medical records to send those questions so that communication on those topics can occur back and forth. And then echoing back to if you hire caregivers outside of your family, sometimes those hired caregivers do accompany you on social outings with persons of the opposite sex, or in social settings where you're most comfortable with your parent being your caregiver. But if you have someone from outside the home, people are professional and they handle those situations differently than a parent would.
Well, and I'll tell you, if this is feeling all awkward to you, life with muscular dystrophy is awkward as well, not just for the person, but the whole family. So don't avoid the awkward, embrace the awkward. It just becomes part of the journey. It becomes part of the story. And if you need to take a PCA, on a date, it's kind of like pretending you're famous and you need a chaperone, you need a bodyguard. I've been in that situation and I've known other people in that situation, and it doesn't have to be a giant mountain unless you make it one. And it can be kind of a fun evening when the third party is helping you with the food and say, look, I have a servant. It's great.
I was just going to add to that. I think that goes back even to our earlier conversation. As Duchenne parents, sometimes we lose our boundaries and we feel like we have to be accessible to our children 24/7, and that's not true. And, I know my husband and I we want to have a conversation right when we get home to talk through things, and we've locked the door, and even with the kids banging on the door, I said, we have got to talk for 10 minutes. You are not welcome to this conversation. And so, I just want to encourage Duchenne parents, you can set some boundaries too, and protect your marriage and protect your relationships with your siblings, and you do not have to be available 24/7, because that sets up a dynamic that kind of, you want to teach your kids boundaries, but if you don't have them yourself, then they're not going to learn them.
And that's with or without Duchenne.
Absolutely. Kathy, I was just going to say that we have under 5 minutes, so I'm going to rapid fire. We've already answered a bunch of stuff, but there's other questions coming in. Somebody asked, what age do you start involving kids in IEP school meetings and learning to self-advocate? As a teacher, I would say as early as they feel comfortable doing it. There are times when I deal with 11, 12, and 13-year-olds. They're almost never in the meetings, and I push for that. I say, the kids should be in here because guess what? It's their education, and they're going to be in here to advocate for themselves. So never mind muscular dystrophy. A lot of parents of able-bodied children are trying to do it all for them, and as a teacher, I don't like that. I say, no, they have to, if they're dealing with a mental disorder or they have attention deficit problem, we got to talk about that. They got to be in here, because it also helps them to own their education. As far as also involving kids, I want to briefly mention, I know it's going to be going through on a slide, but the Patient Adult Advisory Committee, the PAC, we've just launched an app that's available in the App Store and Android, and it's going to be being broadcast all over the world. It's called PAC Chats, PAAC CHATS. You can search up PAC Chats. It's run by us, and it's for anybody with Duchenne, Becker muscular dystrophy. And we have channels within the app that we can talk about all sorts of different topics. But it is also a place for us to come together and not feeling like we're all by ourselves. So we just launched the app. We already have it up to around 50 members. And our goal by the end of 2021 is to have 500 people on the app. So if you're out there and this is for people with Duchenne and Becker. This isn't going to be, again, we're talking about independence here. So if you're listening and you have DMD, BMD, please go download the app. And if you're listening as a parent, have your son go download the app. Don't download it for him. You can facilitate independence by having them do that. Kathy, any quick, or anybody else on the panel, wrap ups?
No, final thoughts. Hey, Bailey, final thoughts?
I just think letting your child ask questions and moving at their pace is going to be the best way. They will ask questions that they want to listen to or hear, and just allowing them to be open will really foster that sense of independence.
Perfect. Rachel, any last thoughts?
I just want to say to all the parents, you're doing a great job. Just take a step forward every day. Don't let this become another mountain you can't climb, but just keep it in the background to be moving that way. And it's for you and it's for them.
and it's okay to let them be more independent. In fact, you all need it. So just keep moving in that direction.
Wise advice. Austin?
Yeah, so I want to add to the part about caregivers. So I think that it's really important with parents because it changes the dynamic. So now I'm not just your caregiver, but I can just be your mom. And I think that with my mom, our relationship definitely has got better from having caregivers where she doesn't have to do all the care for me.
That's pretty brilliant and really good perspective.
Well, and let's face one more awesome silver lining of Duchenne. You, as the parents, you're going to get older, and when your son is in their 30s and hopefully even 40s, guess what? You're not going to be able to do the heavy lifting. So that's a good thing. That's a good silver lining. Plan for it.
Starting early. Christina, final thoughts?
Well, you just kind of hit it. Whether you are young with Duchenne or old with Duchenne, it's never too early and it's never too late to keep working on all of these activities to work towards an adult lifestyle and independent living and making sure you're living your best life.
And finally, last but definitely not least, Pat Motion, final thoughts.
Please send me money.
Not the best thought.
Oh, right. No. Well, again, everybody that knows me knows I'm a goofball. I hope this was helpful for everybody. We all hope this was helpful for everybody. And again, it was an awful lot in a short amount of time. So please, please, please use this as a resource. And just remember that on this crazy journey that nobody signed up for, you are involved with some of the best people that you could be involved with. The PPMD group is really like a family, especially if you're a first timer this year. We really, really care about the whole child, not just the Duchenne and the diagnosis, because it's also about trying to live your best life. So please take a breath and remember to connect with us when times get dark. You are never bothering us, never. You got to realize that we're all on an island, but we're all on the island together. So that's how we're going to get through stuff. Those are my final thoughts. Thank you very much for having me on this panel. I hope this was helpful for you guys.
As a lot of people have said, this is the best club that you never wanted to join, so please don't hesitate to use all of us. Thank you so much to our panelists. Thank you so much for the questions that you gave to us. And now I will send it back to Ryan and Eric in the studio.
Great, thank you so much. You can send Pat Motion money care of Parent Project MD, and we'll use it as a donation. So I want to thank the panelists. We're going to have our brief spotlight on PPMD programming before coming back for a final round of polling and a pop quiz. We'll be right back.
PPMD's Connect program is our family outreach program that is run by volunteer parents like myself and grandparents. This program has been a huge sense of support and strength to our family since our son Brendan was diagnosed with Duchenne. It was really nice to find other families to connect with and realizing that we weren't alone. We didn't have to go through this process alone and that we had help whenever we needed it. And that help came from families like ours who know exactly what we're going through. So it was really, really a sense of comfort for us. And sometimes we get together, like just a whole bunch of families get together or meet up at PPMD events, email each other or virtually meet up on social media. But it's been really nice to have that strength and that hope around us. So if you'd like to learn more about PPMD Connect, you can log on to parentprojectmd.org/connect.
And we're back already. We're going to do our last round of polling and our last pop quiz. Remember, there's a prize for the winner for the pop quiz. I think we're going to be getting up the first question in just a minute. Go to pollev.com/PPMD to access the polling. You can open it up on your phone or on another window on your browser in order to participate in the polling. We have only a handful of questions for this last round of polling, and our first question should be coming up any minute. Again, it's pollev.com/ppmd. And the first question we have here is, in one word, describe one area of care you wish you had more guidance or resources about. We're seeing the word cloud start to come in slowly, and that's okay. I'm going to give you guys some time to answer the word cloud. We see brain, socialization.
Yeah, I think this is another good way for us to kind of get a good pulse of the community in terms of, you know, what you are interested in. And there's a lot of topics out there in care. You know, we got to cover some of them during conference, you know, touching on pulmonary challenges, touching on GI and nutrition. Obviously, a lot more that we could talk about, and I think this is really helpful for us to see kind of, you know, what's important in the community. Whether we do these conversations at conference next year, or we look for more sessions to do with the care team throughout the year, always great for us to see. I see quite a bit coming through, of pretty varied.
Absolutely, it's a whole array of areas of care. Mental health is one of the biggies, and lots of folks are indicating that's an area that they wish they had more guidance or resources about. This helps us really guide us in creating more resources for the community.
No, that's definitely an important area, and something that we also feel as we talk internally, that's an area that we want to make sure we focus on. It's really important.
And it is something we're investing in, and we have a whole program around this year leading into next year, led by Rachel Strader, on Duchenne and the brain. I think it's an area we need more research on, and it's certainly something we hear from the community a lot.
I think see technology up there again, looking for. I think, you know, we see different devices or pieces of, you know, technology that families can use, but I think it'd be great to kind of talk about those. What can they be used for? Where do you get access to them? That's another big piece of that.
Yep, absolutely. Again, psychology.
Psychosocial. Seeing cardiac on there. Again, another area that, you know, we can certainly talk more about.
I see siblings. We just started the siblings group. That's really starting to grow. It's such an area of need for those siblings to have a safe place to talk and connect. They understand each other's issues. We're really proud of that program. That's been growing thanks to Nicole Herring on our team. Okay, no answers are still coming in, but I think we have to move to the next polling question. Thank you so much for all this helpful information. The next question is caregivers only. When your child was diagnosed with Duchenne or Becker, were you given a sense of hope by your physician?
Yeah, and this is, again, we've talked about this in other venues, and I think we're, you know, this is a tough, I'm trying to even think of the words to use, you know, initial conversation to have. And so it's trying to talk with our clinicians about the language used when they meet with families, and seeing kind of your responses, you know, really, I think, informs us. And we know a lot of clinicians as well do tune into conference, and I think it's helpful for them to see the kind of messages.
And they do want to hear. They want to hear this. You know, I also think that for many families, they're in a fog after that first day. And I mean, we're seeing a lot of responses coming in that no, not at all. We're hoping that this is changing, but we need this information as we work with our network of clinics around the country. They need to hear about your experiences with those initial days of diagnosis and that first clinic visit, which is so important. So I think I'm getting the cue that we should be moving to the next question. Caregivers only, again, choose the best description of your support system.
So we just talked a little bit about, you know, facilitating independence, and it's also good to see, though, you know, what kind of networks do all the families have out there. And of course, you know, we have resources, you know, can look for your Connect coordinators that might be in your local area, and help build out some of those support systems maybe outside of, you know, your direct and immediate family.
Absolutely. Absolutely, and you need to take care of yourself if you're going to take care of your child. And that support system is just so helpful for your mental health, your emotional health. And I know not everyone has that, so we try to facilitate that in a number of ways that Eric just mentioned.
And we do hope, as we look towards next year when we hope to be back in person, that you can continue to meet other families and build that network of support, amongst the community.
And we just talked about the young men asking for help, but our caregivers too sometimes need to learn about how to ask for help from their friends and their family. We know that's a difficult thing. Often, sometimes the most surprising people in your circle, in your village, that may not even be family members, are your biggest helpers. But that's okay.
Yeah, and I mean, we saw as it came up within the word cloud, mental health. That's not just for the boys or the women who have Duchenne. That's for the caregivers as well, and making sure that you all have access to help as you go through potentially difficult times.
Absolutely. Okay, we're going to move on to our next one's pop quiz for everyone to take. In ancient Greece, throwing an apple at someone was a declaration of what? This one was for my husband, who's Greek, so I added this into the mix. We have love, war, rivalry, independence. What do you think? Well, don't say it out loud.
Oh, I don't know. I mean, this will be telling of if he ever, threw an apple at you. Was that love? Was that he needed you to just go away for a while?
He's throwing other things at me.
Yeah.
So, he was kind of okay with me leaving on the trip. So I don't know what that says. It's like, oh, you packed my bag. Okay. Things are still coming in. Looks like love is
the winner. Yeah, I guess maybe a lot of people throwing fruit at each other. It's different style. But love languages are very different for different people.
Don't try it at home, though. I think there's other ways we can express this. We don't have to throw things at each other. Okay, let's reveal the answer in just a second. So which of these in ancient Greece was a declaration of what when you threw an apple? The answer is love. Look at that. A lot of people knew the answer. Very good. That's awesome. I think we do have one more question. We want to know in a word cloud, what is one word that describes what you will take away from this conference in one word? I know it takes a second for the responses to come through, but in one word, it might be hard to just do one word, but we always think this is an important way to end the polling.
Yeah, we like to, I think you've done this every year.
I've done this every year, yeah. I like the word clouds.
Yeah, I think it's a nice way to kind of capture a little bit of what the week's been like, and again, how this impacts families.
Absolutely, hope. And so we really do hope that people leave with that sense after conference. See, options, that's a great one. Community, emotional, love, health.
Yeah, all really kind of great words and warm words there.
Yeah. Yeah, luckily, no obscenities or things thrown at me at this point. Pat Motion, I'm thinking of you. Lots of people saying hope, and that's a wonderful
And we see connection and connections, and we hope to, like we said, we hope to build that out even more next year when we get to be all back in person with you. We
absolutely
miss that aspect of conference, for sure.
I mean, the one good thing about virtual, though, you can take your phone outside, you can be on your app.
Yeah.
Like, you can be in the bathroom and do this. We don't care. I mean, it's not our business. It's between you and your family.
Yeah, we get to reach a lot more folks, which is really
true.
important.
It's a lot more educational than liking memes on Instagram, too. So looks like hope is the predominant option here.
Seeing resilience, it's another really big one and an important one, especially with the way we've been through the past year. So it's great to see everyone still being resilient.
Support, health. It's great. I mean, these are all things we want to continue to have people feeling the end of conference. And I cannot wait to be back in person. Next time we are going to be in person is the Advocacy Conference next year. We hope people will take us up on coming to Washington March 6, 7, 8. Yep. So Should be a really good opportunity for us to have some reunions. Support future independence. Emotional, I'm quite sure. All the social interactions we've had, the Zoom social nights, I've heard a lot of positive feedback from that. I know everyone's so sick of Zoom, but I think in this context, when you're talking to people who get it, it's a wonderful thing, and you're probably you're okay to be on a Zoom in that situation, aren't you?
No, you're fighting your community. You're able to share things with people who understand the same type of things you're going through, and as well can share their experiences, which is always really important.
Absolutely. Okay. I honestly could keep this going. I love watching these come through, and I thank everyone for doing the polling. Eric, what's up next?
So I think next up we are going to share the results of our daily challenge winner.
Yes, we've got our daily challenge winner. So this is the Forrest family from Virginia. You've won a $25 gift card, and then we have our 4-day challenge winner. This is Cole Terrell. So we went back to the daily challenge winner. Sorry, this is the Anything Goes. This is the Forrest family. Awesome job. And then this is the 4-day challenge winner. A $50 gift card. Entered all 4 days. Cole, you're my hero from Smithfield, Virginia. I think that I love this part of conference. I thought this was so great.
This was very fun.
Everyone who participated, we cannot thank you enough. It was a real cool addition to add to the conference.
Yeah, it was great to see. Love your summer style.
Absolutely.
All right. I think with that, we are going to move on to our final panel of the day, which is going to be on quality of life and awesomeness. So in this panel, we're going to be joined by some caregivers and care specialists who are going to discuss awesomeness. And with that, we are going to hand things over to Pat to get this started.
Hi, welcome everyone, and thank you for joining us. I just want to introduce this panel because you have 5 amazing people to talk about this, but we've been working on language. Language is really, it's always been important to me, how we express ourselves, how we talk to each other, how we think, the stories we tell ourselves. So for the last, I think about 8 or 9 months, we've been having a small group of people get together and talk about their roles as moms, the stories they tell themselves, the language they use, and the interactions they have with healthcare professionals, with their partners, with their husbands, with their children. And we've emphasized language. In fact, we also talked about metaphors, about how we as moms have all these different roles. And no matter Duchenne or not, you have all these different roles as a mom. And Alpa came up with this metaphor to describe herself as the kitchen junk drawer, that there were so many roles, and when other people didn't want to do them, they stick them to her in the junk drawer. So we want to kind of explore that, because I think at the end of the day, as a mom, as a caregiver, just as a woman, you're going to be constant in your life, and things are going to change around you. For some of us, it's a diagnosis of Duchenne. For others, it's different life experiences. So this is a panel that's going to talk about what they've learned and what they're thinking about. So I'm going to send this over to you, Kathy.
Thanks, Pat. I do think language is so important, and the things that we tell ourselves, the things we tell our children, the things we tell our care providers, just the things that we say and the words that we use, and the mental images that we have, I think, make a big impression on how we live in our day-to-day life, and how we present ourselves to ourselves, and how we present ourselves to others. So before we get into this conversation, I wanted to introduce, or have the panel introduce themselves. So I'll just go through my list. Kana, do you want to go first?
Sure. Hi, everyone. My name is Kana Maraki, and I have a son, Sammy, who's 10, with Duchenne.
Mina?
Hi, my name is Mina Scavina. I'm a neurologist at Nemours DuPont Hospital for Children in Wilmington, Delaware, and I've been able to participate with all these women over the past few weeks, and it's been an honor.
Thanks. Alpa?
Hi, I'm Alpa Kushalani. I live in Fayetteville, Georgia, and my son Krishna is 13 years old with DMD. I have 2 other boys, Ishan, who's 10, and Bihan, who's 7 years old.
You do not look like a junk drawer. Maria Jose?
Hello, everybody. My name is Maria Jose Contreras. I am originally from Chile, but I've been living in the US for the last couple of years. I have two boys, Franco, he's 7 years old, and Julian is 4 years old, and both of them have Duchenne.
Thanks. So we've, as Pat said, we've been talking for about 6 to 8 months and talked about a lot of things, but I think starting with the junk drawer, we do have a lot of roles, and it's really hard to separate what our role is from minute to minute. And people depend on us for so many things. I remember one time I was miles and miles away from my house, and my husband called and said, "Where are my car keys?" How am I supposed to find your car keys? I'm 50 miles away. But we walked through it and we found the car keys. So how do we start to separate some of those roles and how we feel about ourselves in those roles? Jump in. Kana, I'm going to call on you. So you got to start.
Well, we've, I believe in our conversations, we talked about all the roles that we play, right? And, from caregiver to wife, to partner, to, coworker, and how our lives are so full, and how do we start to define what is important to us and the roles we want to step and lean into more versus the ones that we maybe need to take a more of a back role in. And maybe with me stepping back a little more, I'm finding that people step in a little bit more. My children become more capable and more self-sufficient. My husband will take care of some of the things that I would otherwise, being the typical junk drawer in my family. And so I think it's really helped to really define the roles, but also think more clearly about what are the roles that are truly important, truly additive to my life and the life of my family.
Very well said. I'm kind of, picking some points from the discussion that happened right before this on independence. And I'm thinking when we think of our role, we are putting ourselves so much in the center of everything that's happening in our house that we are involved, and we take the role of being the care provider, of being the cleaner, of being everything, that it kind of comes back to us. So, today as I listened to independence, it was it's important for us to start delegating some of the roles that we are doing so that we don't get a call of where the key is, or, Mom, where are my swim trunks? Or, where is my pen, for all that it matters, and making our kids more independent. So us, trying to show them the way, lead the way versus, doing it for them. So that's an area that I believe we all can improve on and, help them have better roles and start participating in some of the roles that we do.
One of the things, go ahead.
Sorry. Yeah, and it's about the roles, but also in a smaller scale, it's also about the activities and what we devote time to. So I think there's also the issue about balancing what we do for others and maybe what we're able to do for ourselves. I'm not the one to speak about this because I really think I'm working on it still, and that I need to do a lot of work. But I'm aware of how much after the diagnosis, the variety of activities that I did before have narrowed, and I've concentrated more in the care of my family. And so I think it's really important to really have this view and to acknowledge how difficult it is to step out of that position and to pick up some of the roles and also the practices, the activities that we did before. In this small group that Kathy and Pat was just talking about, we did a list about all the things that we stopped doing after the diagnosis and that we'd like to pick up again. And mine is really a large list. So I don't know if I'll make it to pick it up, but for sure I know that it is important to take back some of that time for myself. Also, in order to care for others in a better way, in a healthier way.
Yeah, I think we take on so many things that it's really hard to take on all those things without burning out. So how do we prevent, and one of the ways to prevent burnout is to try to take care of yourself a little bit, but Maina, how hard is that? I mean, without even being a mom of a child with Duchenne, it's hard for everybody.
Yeah, I mean, I think it's a very good question, and I don't know that I necessarily have the answer, but I think we can almost feel it when we are just surviving by doing the things that are asked of us all day long and going through a rote day versus thriving and really feeling that we are improving the lives of our families, our patients, ourselves. And so I think it's really important, and this conversation, I think, has made us all more aware of that, because it's easy to forget that for sure, for everyone, for mothers, fathers, providers. And so prioritizing what are the most important things, having people take responsibilities, even the boys, you know, when we have patient portals. And as boys become teenagers and have questions, I'm always thrilled when they will send me a question themselves. Hey, can you renew my prescription? Or what do you think of this? And so delegating those responsibilities may be very helpful and empowering to those around you.
And you know, the conversation reminds me of how we talk about, you know, I don't know if, I think everyone's felt at some point in their lives that they've sort of missed the moment. They've missed an important moment because they've been thinking, in my case, about the laundry list of all the things I have to do in my life. And, you know, and I've missed this beautiful family moment of my daughter telling a funny story about what happened in her day. And so I think we talk also about trying to be more present and really being there and not getting carried away with all the things that we feel responsible for and the things that we're sort of carrying on behalf of ourselves and others.
Right. And one thing that I do feel is we all are being so hard on ourselves. We just need to stop being hard on ourselves. You know, there are already enough people in this world who do it for you. So, you know, we just want to, I mean, we'll make mistakes, and making mistakes means that we're trying. So we're trying to be better for the child we are providing care for our family. You know, it's always a balance between us, our children who have DMD, the children who don't have DMD, the time for our husband, you know, jobs that we're doing. So all I can say is, you know, it's like sometimes all we need to do is just say to ourselves, like, I am enough, you know, as we were talking, just I am enough, I am doing my best. And, you know, I went through a program some long time back about, you know, just mindfulness and meditation, and it made us write. We had to write, like, I absolutely and completely forgive myself for, and then write all the things down. It kind of was liberating for me in some way. So maybe that's something we could do, you know, once in a while, and just, you know, forgive ourselves for being so hard on ourselves to begin with. And that's probably the first step of self-care.
It's hard to give ourselves grace when we need to do that, isn't it?
Mm-hmm. Absolutely. And sorry. Yeah, I was thinking that this awareness that we've gained have pretty much emerged from this collaborative group. So also like to say that I think that to really be in this working process of being always present, or trying to be as present as we can be, or of taking care of ourselves, it's so important, like the network of support and having other people where you can, to whom you can speak with, and also with whom you can accompany yourself in this process. Because it's like, I feel like without this conversation with the small group, it would have been much more difficult for me to arrive to this standing point where I feel I'm aware, at least, of a direction or a path that I'd like to follow, even if it's still all like in front of me. It's like an ongoing process. It's not something that you can decide and then be always present or whatever. But it's been very, very important to have like this village with me. To really do this work, because it is a work.
One thing I think women struggle with a lot is guilt. I don't know many men that struggle with guilt, but maybe they do. But I think women, and especially moms, struggle with guilt. So, you know, when you're not taking care of your son, you're feeling guilty. And when you're not taking care of the siblings, you're feeling guilty. And when you're working, you're feeling guilty. And when you're not working, you're feeling guilty. So I don't even know where to start with that. Anybody have any thoughts?
I don't let my husband go guilt-free. I think I take him on the trip with me. So, so I don't know about that. But yes, we all are, as I was saying, we just feel guilty for not doing enough. And that's why just telling ourselves, I am enough, I am enough is such an important part. And I cannot thank Mina enough for our conversation one day when she said, the time you enjoy wasting is not wasted time. Like we feel so guilty taking any time for ourselves, and we feel it's wasted time. We could have been doing something else. And I have taken that to my heart, and I say that every day to myself. The time that you enjoy wasting is not wasted time. So take time for yourself and stop being guilty.
Yeah, thank you. Last night I was able to join some of the mothers in discussion, and one of the mothers said something that was very interesting that I thought to share. And she's a mother of an older young man, and she said, I wish someone had told me at the beginning that, in a sense, this is a marathon and not a sprint, and that you need to be in there for the long run. And I'm quite exhausted now and need some time for myself, where maybe I could have done this all along a little bit. And so it's very helpful, I think, for myself as a provider to hear these things, because it's often hard to know where people are at and what are things that are really going to make a difference in clinic. And so I think that that's very important. So you don't burn out. So you have this ability to stay in there for the long haul.
As they always say, you can't, pour from an empty cup. So we got to fill ourselves up, and especially for our boys, because there is a lot that they deal with and they will deal with, and they mostly look at ourselves and our reactions. So when we are in the provider's office, in this meeting and this long day that we have at the MDA clinic, more than the provider, they're kind of looking at us and our faces and our reactions. So, one of the great things that we spoke about in our group, and that was like always keeping in mind also what you're proud of about them. What are the things they can do? So when we go into the meeting, instead of only focusing on what are the things they can't do or things that have changed, also try to think about the things that they are able to do. So, what are you proud of that they are able to do? And that helps send a positive message to the child as well. Yes, I'm able to do these things. My mom's not coming and saying, oh, he used to be able to climb steps independently, but now he's not. But instead, oh, he was, he's still walking around. So focusing on that, the positivity, along with not losing focus on what you need, will set a good mindset for the entire conversation. That was one of the important learnings that we had from the discussions we did in this small group.
Yeah, like the best interactions that as a group we found with care providers specifically was when we could engage with them, like from a personal point of view, personal meaning that our son is not only the diagnosis, and this comes from both parts, from our family perspective. So what we tell to the provider, as Alpa was just saying, is not only like the deficits or what's not working, but also like other things. And as Alpa was saying, I was thinking, yes, what they can do, but not only referring to their motor skills, also like if they are good at math, or if they love reading, or if they did an awesome story or wrote an awesome story at school. So how we can really we as family and as caregivers like install another language in our relationships with the healthcare providers, so that the conversation can pivot from this patient medical way of looking a person to really looking from more an integral point of view, and considering like as like all the aspects of our sons and of us as family of them.
Yeah, I don't think anybody wants to enter a room and just talk about what's wrong with them, right?
Yep.
So Mina, you had mentioned that you changed the way that you started to do your interview style based on conversations.
Yeah, and I've had some mothers who've come and really started talking, clearly giving us the reason they're there, but also telling us many of the positive things about their child. They may love horseback riding, and that's important to them. So the goal of being here is to understand how we can address the problems so that they can accomplish that. And so I think what your goals are, what the child's strengths are, what you want from this interaction. I think those are ways that make the relationship you have with your healthcare providers, everyone from the physicians to the therapists to the social workers, everyone, more personal and real. And so many of you go to clinic and it can be very chaotic, confusing. And so maybe having an idea when you go in there, these are the things I really want to relay about my child and sort of my goals. And, you may have the provider that they have their own agenda, but together maybe work on how you accomplish those things together. So I love when people tell me more about their child. I mean, the other day, I walked into the room of a 6-year-old and the first thing he said to me was, "I pooped." And, it suddenly, brought a levity to everything. We started talking about constipation, so rather than ignore that and say, "I'm here today to talk about this with you," take that and run with it. These are the things that the children want to talk about. These are the things the parents want to talk about. And, my saying, "All right, this is awesome," it just really took a little bit of the stress out of a situation that was very stressful for a family. So, I mean, there's many styles in medicine, but you can also have an impact by what you bring to the visit.
I think that conversation just about sort of, the different perspectives of the caregiver, the patient, the provider, and what it also made me think was that previously I didn't really think so much about like what is important to me in a provider. And so I started, me and my lists, I started to write things that I'd never really considered, like, what are the values that we hold as a family? And those are the things that you do communicate to your caregiver and sometimes to your provider. And sometimes it's not, sometimes it's not a match and you can make it work. And you have to look at your provider also as a human, right? Sometimes, and Mina, we've talked about this where sometimes, doctors are having a bad day, or your PT may have something going on at home where they're distracted or they're stressed or whatnot. And so we can't, it is a relationship. And the more we think about what is important and what are the things we value, in the relationship really helps us kind of achieve a relationship that is more in sync and more clear and have clear expectations of each other of like, well, this is really what is important to me. And hopefully I think that does result in better care and better relationships.
As we were doing this entire small group, we had the opportunity to listen to and work with Rachel Callender. If you guys have not had an opportunity to look up her TED Talk about the Super Baby Project. She lost her baby, her child pretty young at 2.5 years, Evie. And, it's amazing how she said that, every time, obviously, since you have a progressive disease, you're looking at things that you are losing. But how can you focus on what you still have? So she had something called the awesomeness report, where it's like, she's like, as I sit and fill all these forms, like we all do every 6 months, or here, is your child able to pick a gallon of water? Or, in the last week, was he able to do this, that, or not? So, she said it was hard. So every time we, answering things that went to the negative. So she came up with this awesomeness report of all the things that she was awesome at doing. And that, if you look up her book on the Super Baby Project, it's just amazing, something very empowering. And that made me think about, all the awesome things that Krishna can do. And I was like, even though some of the things are not, positive in the positive sense, but it's like, he can give his, he's given his hand, 8 times to be stuck before they got the wing. And, that is a superpower that I guess most of our DMD kids have. So it's like finding what is your superpower, you know, like how teachers say teaching is my superpower. So resilience is definitely a superpower that all our kids have. And, you know, we have to keep looking for that silver lining, as we all say, right, at all times. And we're fortunate that language is one way that we can make that happen for ourselves and for our kids and for our providers. That's the binding factor.
And language is built in conversation. So as Kana was just saying, it's something that it really, it's like finding a point where you can meet, like, or make the encounter of two worlds, the one of the provider and the world of your family or of your child. And as a family or as a caregiver, for me, it's very important to express, maybe there are some providers looking at this, at our conversation, how their language also influences a lot how we can think of our child. I had this very moving experience when my older son Franco was just diagnosed. We were still in Chile, so I asked for a meeting with the school teachers to let them know about the diagnosis, and I just wrote an email with, my son was just diagnosed with Duchenne muscular dystrophy. I'd like to meet with you and speak how we can better support him. He was in preschool, and we had this meeting, and they came into the meeting super prepared. They did like the right questions. It was evident for me that they have done some research by themselves, because I didn't have to explain so much to them. They already knew what we were talking about. And also they were like very empathetic and sympathetic, but it was not like emotional in the sense of looking us or looking Franco as a victim or something. It was like, okay, let's work together, how we can make his life better, how we can accommodate to what he needs. And at the end of the meeting, and this is something that really has marked my journey with the diagnosis of my sons. At the end of the meeting, the principal, he was in the meeting, but he didn't speak at all during all the meeting. And then at the end, he said, well, we're finishing this meeting. Thank you very much, blah, blah, blah. And I didn't know if you noticed when you came in, that in the playground, we just built a bench because we realized that there was nowhere to sit. So now Franco and his friends will be able to sit if they are tired. And not only the gesture of building a bench, like from one week to another in the playground, but also that he said Franco and his friends. At that moment, I was just had received the news of the diagnosis. And one of my most dreadful fears was that he wouldn't be able to participate in activities with other kids, that he wouldn't be able to make friends. And this principal of the school told me, Franco and his friends can sit on that bench. And that really like make like a setting, a mind frame for myself on how to think about Duchenne and how to think about the possibilities of my son. So this is just a message to school providers or healthcare providers that also the way that they frame things are really important to us.
Yeah, I love that story when you told us, Maria Jose, and I wish more could be like that. This is not an excuse, but I think sometimes the words we use are very, you know, they're made from textbooks. They're made to communicate between medical providers. And so when we use them in clinic, I think sometimes they don't have the human touch we would like them to have. I love when providers can make a metaphor for something, for an explanation. And I think those are very effective, but realizing too that it's been a long history of those words being used, and sometimes we don't always have the right ones to replace them with. But I think that's where we can change things, by the words we use, the things we say, even the word in this space of mutation. You know, very commonly the word mutation is used. And I remember the first time I spoke to a family and I said, well, this was a number of years ago, but the mutation that we found, and the father immediately stopped me and said, you use that word with people? And I mean, I had always been taught that's the word we use, right? And he said, that's not a great thing to relate to a family. So it's just made me mindful of using that. And even when we did these videos, we tried to use variations or variants or different fingerprint in the DNA. And so it's going to take a long time, but these kinds of conversations I think will be helpful. And I think sometimes if you hear certain things, to tell the provider, that's a very hard thing to hear as a parent. They may not even be aware.
I think as providers too, we sometimes will default to language that we're comfortable with because we're distracted, and you don't, or many times distracted, and with the next 3 people that you have to see and the labs that you need to discuss and the things that you're trying to do. But I think trying to be present in that moment with that family and with whoever it is that you're speaking to, your child, whoever it is just so important, and helps to pick the most appropriate words and the most that will help to get the message across the best.
And kudos to the dad for just mentioning how that landed for him, right? Because, you know, it's hard. It is hard for anyone to deliver hard news. And I think of providers as having this really very difficult job where they do it repetitively. And, you know, it's a very natural thing to start using the same words because it's easily accessible. You're already nervous. And so, you know, you may not be choosing those words exactly the way you would have if you, you know, in a different circumstance. So this does, you know, this is something that we talk a lot with my son about. And when we went in to go to talk to his school and his classmates, Sammy was pretty adamant initially that he really didn't want to share his diagnosis with his classmates. And when it was the right time, much of our conversation with the PT who went in and with the teachers was to talk about, let's talk about the language, right? Are we talking about a disease versus a condition? Well, disease makes people uncomfortable. Is it, you know, is it contagious? All these things. And we talk about special treatment versus an accommodation, right? A special treatment for young kids is like, whoa, he's getting special treatment. And talking about like learning tools. And when we start to use this language and show people like this is the language that is makes Sammy feel like he is part of the school and just as much as any other child, I think it just kind of helps everybody if we're the ones within the community to give people the words to say like this is neutral, this is loaded, this is makes people feel uncomfortable or less than. So I think language is incredibly important to teach our boys and men and the greater community.
So beautifully said, Kana. That was amazing. I think for our kids, yeah, it's important instead of saying you can't do this, you know, let me help you do this. So it doesn't start with that negative where they're thinking, okay, you know, I'm not able to lift this milk, so let my mom pour it for me, you know, or anyone else. So letting them, you know, not constantly be reminded of the things that they have difficulty doing versus, you know, trying to present it more positively. And also, you know, it's cliche, but, you know, you'll forget the what people said, but you'll always remember how they made you feel. So sometimes we may not have the right choice of words, but if you have compassion, which Pat in one of our conversations said, love without pity, and I've taken it to heart again. You know, if you can show them the true love without really feeling the pity for them, then I think we'll all be okay.
Absolutely, and how important it is on the one hand to speak and to be aware of the language, but also to be able to listen. And this is also so important with our boys or with our sons in the sense that many times, even if my sons are still young, they say things that for me are very difficult to hear and that may break my heart. But I think that the work we need to do is to make the space for them to be able to express like these feelings. My youngest son, Lian, he's 4, and they saw this Disney movie Soul that's about souls. So they started to speak about death. And he told me after the movie, I will die soon. And my mom was there, and how she reacted was like, no, what are you saying? You're a kid, that's nonsense. But I really tried to listen what his fear was. So I opened the question. It's like, why do you think you'll die soon? And he told me, because I have wounds. And I said, I don't see the wounds in your body. And he said, they are inside of me. So it's a very difficult situation. It's heartbreaking, and the easy way to go is like, okay, no, everything it's okay. That's not bad. We really need to stick in there. And then we started to speak about muscles and how they break and what it means to have Duchenne. And afterwards, we spoke about it. He was like very calm, and he like a minute afterwards was playing. But I really think it's important, and it was important for me to be there present with that fear with him and not discharge him and not, not listening to him. So I think that on the one hand, it's language and what words we use, and on the other, it's how we are able to listen so that we make space for them to express not only what's good, but also what the fears they have.
Well, I think one of the things that you all have are the superpower to be able to listen and to give space to those things that are important to your kids and important to your families and important to your patients. So I appreciate every one of you, and I appreciate all of the work that you've done on Wednesday nights. I appreciate you being here today. And now I challenge you to go forth and share those superpowers with other women. So thank you so much. And with that, I'll kick it back to Ryan and Eric in the audience or in the studio.
Thank you so much. What an incredible panel, huh?
That was great.
Really, really incredible. We want to now just take a quick look at some of the photographs that came through for the challenge this week and highlight some of those photos so everyone can see. We want to thank everyone for participating in this. It really was a fun addition to conference, and we're grateful to everyone for adding, moments from their life for us to see and for the community to see.
Yeah, especially when we can't see each other face to face. This is such a great way to see everyone, and such a fun way for that to happen.
I loved all the themes. All the themes were really fun.
Yeah, these are all great.
To get involved.
Seeing really all the wonderful families.
And we'll be sure to post these on social media so everyone can see them. And I think it's something we should consider doing more in the future. It's a great way to get everyone involved. Oh, this is one of the winners.
Yeah. There's another one. I loved the inclusion of the chicken in all of the all of the different ones that we saw there.
It's a great way to get the family involved and the pets.
Yeah.
Look at that.
Always happy to see a dog or a cat. I like the sunglasses.
Yeah, it's pretty cool. Oh, that's awesome. Thanks everyone for wearing your PPMD swag. Gave us a lot of great pictures to use in the future too.
Mm-hmm.
We love seeing all these pictures. We love seeing you in person more. Not enough hats. I think those are the sunglasses that we included.
Oh, okay, yeah.
Yeah. Oh, that's awesome. I love a good bow tie.
Mm-hmm.
Oh. There's Chris and his family. Pictures with your pets. It's really, really wonderful.
Or just the pets themselves.
Or just the pets themselves. Those outfits.
Those are not Jedi, Ryan.
No.
Just want to make sure that's clear.
Yeah, I got a lot of hate mail for that. People are very serious about Star Wars, apparently. But that's okay. My apologies. Okay. So thank you so much to everybody for participating in that fun activity with conference. We do want to announce the polling winner. Caitlin Watson, you are our final pop quiz winner. You'll win $25. We'll send that on to you and all of our pop quiz winners. Thank you. You were the smartest in the bunch, so we're going to send you your prizes. So thank you for joining us this week. We know it's been a lot of content, a lot of your free time, and not the same as in person, but I think it gave the opportunity for those who may not be able to travel to conference to have the ability to connect. And we're going to find more ways to do that even when we are in person for conference next year, sort of a hybrid approach. So we hope your family enjoyed the week and feel more informed and empowered at the end of the day. And I want to thank the Dudley crew here, who are incredible. Dudley Digital Works. We could not do this without them. I can't say enough good things.
Oh
about
yeah,
them.
you guys made our lives
They've been very
tremendously
patient,
easy.
very patient as well. So Eric.
Yeah, and so we do hope that we will get to see some of you in person. So we hope you will all join us at the Advocacy Conference next March. We'll be meeting on the 6th, 7th, and 8th. And then looking forward to conference in 2022. So we plan on seeing everyone there. We're going to be back in the Phoenician in Scottsdale, Arizona.
Everyone loved that space,
Yeah,
so
it was
we're
great.
excited to go back. And last but not least, we have our closing remarks from PPMD's founder and president, Pat Furlong. Pat, please take us away. Goodbye, everybody.
This is always bittersweet for me. It's always the end of being together as a community this year virtually. Thank you for attending this year's conference. I wanted to close with some reflections on the past 4 days. First, we have reasons to celebrate. We heard about the progress of approved therapies that have confirmatory trials underway. And we celebrate that more options are on the horizon. We're thankful for the companies working to deliver safe and effective therapies for everyone living with Duchenne. But we also recognize the challenges we all face. We will continue to stress the urgency to all companies and to the FDA to speed the development of therapies, including those follow-on exons, for all those that have been waiting. The truth is that even with the incredible progress, drug development is painfully slow for families who've waited to see their exon of interest in the clinic. We need to pursue solutions for every imaginable angle. That's why we're trying to develop tools with the consortia such as the D-RISC and with CPATH and CTAP and MRI, which you heard about from Dr. Vandenborn, and biomarkers from the University of Florida and Dr. Spitelli. We also appreciated hearing about therapies developed for non-ambulatory and those diagnosed with Becker muscular dystrophy. To those companies, we say Godspeed. We also want to continue work with companies that have approved therapies on the collection and release of data. Data is everything. Clinicians need this data, families need this data, payers badly need the data, FDA needs the data, and you heard Dr. Smith say, he and other clinicians need this data to explain and support shared decision-making, to know the right time, the right dose, and the right therapy for every person. But most of all, our families deserve this data so that they can make the best and most informed decisions possible for themselves and their loved ones. To make further progress, we're updating the Duchenne guidance, so we'll provide a roadmap for companies in this space and for the FDA, so we can learn from past trials and run new trials more efficiently, including new strategies like second-generation exon skipping, as well as gene replacement and gene editing. Finally, cardiologists' opinions are changing to treat very early. That's why we're including this new section in the guidance. We continue to fund the New York State newborn screening pilot to diagnose early, and we're collecting real-world evidence from our community through the registry to have data directly from you about your experience. Tackling Duchenne from diagnosis through drug delivery is the only way forward. So my message to you is, we hear you. We will continue to support those critical pieces in our drug development ecosystem to push for therapies with your input and preferences at the center of it all. We know how hard it is waiting. We know nothing happens fast. Please know we also amplify these sentiments every chance we can. We appreciate all the work of all the companies, the academics, researchers, regulators, and our message to all of them is the same. Move with expediency, communicate regularly, openly, and honestly. Collect the data and level with us. The community deserves nothing less. So with that, the 2021, I'll get it right, conference is closed. And we're all anxious to gather with all of you once again in 2022. This is in Scottsdale. We will see you very soon, and thank you for joining us. The conference is now closed.